If it had been otherwise, I would have published a different perspective in Science, and we would have published a perspective or a couple of papers showing that fuck was a lab.
What's up, you scholars of enlightenment? I hope you've had a fantastic week and have a wonderful weekend in store. The origins of SARS-CoV-2, the virus that causes COVID-19, are obviously of great international concern and interest. Regardless of the specific origins of the virus, changes in our collective behaviour, customs and policies will be required to head off future pandemics and the massive disruption that accompanies them. 2 new peer-reviewed papers from a leading team of virologists and scientists have now spread further light on the origins of the virus and explore multiple lines of evidence that point to the Huanan seafood market as the ground zero of the outbreak and wildlife trading as the most likely cause of viral spillover into humans. To help me break down those papers and understand their results, reasoning, and implications, I'm delighted to welcome 2 fantastic special guests. Firstly, Professor Kristian Andersen. Kristian is a professor in the Department of Immunology and Microbiology at Scripps Research. Over the past decade, his research has focused on the complex relationship between host and pathogen. Using a combination of next-generation sequencing, fieldwork, experimentation, and computational biology, he has spearheaded large international collaborations investigating the emergence, spread, and evolution of deadly pathogens, including SARS-CoV-2, Zika virus, Ebola virus, West Nile virus, and Lassa virus. His work is highly cross-disciplinary and exceptionally collaborative. Secondly, Professor Michael Worobey. Mike taps into the genomes of viruses using molecular and computational biology to understand the origins, emergence, and control of pandemics. He has made discoveries pinpointing, for example, where, when, and how HIV originated and spread worldwide, and how influenza pandemics, including the intense 1918 pandemic, emerge and kill large numbers of people. His current research includes SARS-CoV-2 genomic epidemiology and evolution from local to global scales, work at the intersection of viral evolution and immunology with SARS-CoV-2 and influenza viruses, influenza vaccines, and pandemic preparedness and prevention. So who better to help me break down these papers and understand their implications? So first question I want to get into is, when did you guys get interested in the origins of COVID and potentially finding the origins? What motivated you to start looking into the origins? Because it has become a little bit of a shitstorm. So let's start with Kristian. Why did you get involved, apart from Tony's big suitcase of cash, obviously?
Yeah, I mean, the thing is, understanding the emergence of outbreaks in general is something that's been the focus on much of our research for more than the last decade. And we've done this with various different viruses, like Lassa virus, for example, understanding that in West Africa, Ebola virus in West Africa also in 2014, for example, and also Zika virus here in the Americas, and especially the emergence of that virus, both in the Americas, but also in the United States. So these types of questions is something that, you know, my lab has a lot of focus on. Yeah. And here was a novel coronavirus that had just emerged in a city in China. It looked a lot like SARS-1 in terms of the emergence, and the virus itself was quite closely related. So I got interested in that. And then, like everybody else, I think, I noticed, you know, I was aware of the Wuhan Institute of Virology. I was aware of the type of work that they did there. I was aware of the recombinant work, for example. I was aware of the isolation, for example, of novel coronaviruses. And that got me concerned, to say the least. And that's why I thought a priori, as a hypothesis, saying like, actually, here we have an example in which we shouldn't just automatically assume that this is a natural emergence of a novel virus, because we actually have a potential alternative explanation here that we also need to consider. So that's what got me interested. And then I'll say, did some early analyses. Again, I am an expert on like viral emergence and evolution, but not on coronaviruses specifically. So very early analyses, had a quick chat with my colleague Eddie Holmes, who has been doing this his entire career, and explained to him some of the concerns that I had. And then we took it from there. Eddie had, similar concerns, and then we started looking at this much further. And all of this was, of course, in January, end of January 2020.
It's interesting you say that, because that sort of initial uncertainty that maybe you had and some others had has now kind of taken on a bit of a life of its own, that people don't seem to be able to let that go, that the process of science has moved away from that. Any comment on that before we move to Mike?
Yeah, I think it's important to understand that in the absence of evidence, right, this is a reasonable hypothesis that we certainly need to consider.
We were both concerned about it, Philip and I, although like many probably are surprised to hear that as well.
Yeah, I think again, when you're in a situation in which everything is new, right, and everything is chaos, and you literally have no evidence, you need to continue to look for that evidence and saying, like, actually, what does that evidence tell us? Some of that evidence then emerged very quickly, and I will say there's lots of early papers from China in, for example, the China CDC Weekly, describing early hospitalizations. The fact that they were picking these up at hospitals, for example, was being described. Of course, more details around the market, the statements from the China CDC itself, as well as in publications. And then really importantly, also things like, well, here are these pangolin coronaviruses that have exactly the same receptor binding domain, a very similar receptor binding domain, I should say, as SARS-2, was a bit of a revelation, right? But we also have to remember, though, that they're just a priori that all of this being associated with a wet market, exactly what we have seen previously. The idea that somebody could sort of just randomly go out and pick up the new virus, the next pandemic virus in a bat somewhere in China, right, is by itself extremely unlikely, compared to the multiple, wet markets you have all over China, and animals going through these. And it's not just China, I should say, it's much of the world, right? Yeah. Is that the risk of these types of emergence events with these viruses being very diverse, and there's a lot of them trying to jump into the human population all the time, but are unsuccessful in 99.9999% of the time, right? Is that all of that changes once you have just the right setup, which unfortunately things like wet markets, right, but is one of those examples.
Mike, anything to add to that?
Yeah, so I've also spent a long time, better part of 25 years, trying to pin down the origins of various pandemics, including HIV, Spanish flu, things that could become pandemics like H5N7 and H5N1 and H7N9 viruses that are repeatedly jumping in from birds, and really trying to use the genomics of the virus and the phylogenetics to learn what we can about what makes a pandemic happen, and what we can do to prevent them, or at least mitigate them. And my perspective on this has been also informed by my own lab's work. So I, at the time that this pandemic began, I had a fantastic graduate student who's now postdoc, Brendan Larson, who was out in the field in Arizona collecting samples from bats to look at paramyxoviruses. So, we have a freezer full of bat samples, and my, I was never, I never had a moment where I, for example, thought that the furin cleavage site required a non-natural explanation. I just, as an evolutionary biologist, it just, evolution can certainly deal with things like that pretty adeptly. But what I am concerned with, in my own lab, is the possibility that someone has a bad day and is processing a sample and touches a glove to their eye and just screws up and all of a sudden you have an infection. And so I've been, amongst the most open scientists to this idea that at least some form of lab incident, and again, I've been focused on just an accidental infection, maybe even with a virus that hasn't been characterized in the lab from a sample, that no one knows has a SARS-related virus in it, could have infected someone. And so I actually initiated this sort of fateful letter in Science Magazine, which I initiated it in the sense of suggesting to Jesse Bloom that, maybe it would be a good idea for scientists to just say, hey, let's keep an open mind and consider possibilities, because at this point we hadn't heard from Xiao and Xiaomin Zhou in their scientific reports that, for certain, these animals like raccoon dogs and foxes and whatnot were even for sale at the Huanan market at the relevant time. I hadn't done, I had a perspective in science where I drilled down and really convinced myself that the apparent pattern where a large fraction of cases early on really were linked to people who worked at the market was a real phenomenon, rather than some sort of ascertainment bias where people were like looking at the market and finding a bunch of cases there. That I hadn't done that. And so at that point I was much more open. The evidence was in a very different state at that point. What I wasn't really involved with is drafting that letter. And in retrospect, I was pretty naive about how that letter would land, and shouldn't have been, and regret the tone of the sort of, open your records, which is essentially directed at Shizhong Li's lab in the Wuhan Institute of Virology. And at that point, Shizhong Li was already on the record saying, listen, we didn't have this virus. The people in my lab were tested and were not positive for SARS-CoV-2 in the spring of 2020. And so essentially that letter kind of accuses implicitly Shizhong Li of being a liar. And to me, that's a great regret. And that letter then took on a life of its own in terms of contributing, I think, to Biden's 90-day intelligence review, and really taking this theory that was, my intention again was, this is a sort of 1% possibility, maybe at most, but let's not, as scientists,
forget the 1% chance. Yeah.
Because you have a huge impact, even if it's a small probability. So it's still important to understand that in terms of risk.
And I'll say,
anyways, that I think you get the picture. That's where I came at this from, and then from that point on, I have been, along with Kristian and I kind of came at this from, we were doing sort of independent work where he was really focusing on what's going on within the market, and I was focusing, largely on my own at first in my basement for month after month after month at what's going on in Wuhan spatially. And then we kind of realized that we really need to just put this into one package. Meanwhile, I was also, but Kristian and I were also working on this other related project led by Jonathan Picard and Joel, and that it was all part of the same project, really, but became its own separate paper.
Yeah, so I think we're jumping a bit too far ahead. So maybe we just do a quick summary
I think that
questions.
journey, Philip, is
Yes.
really, really important for people to understand who are watching, right? That what's been said even today by some of our favorite book authors has been that this is, a cabal of self-interested scientists who were never interested in a lab leak hypothesis. They wanted to go out and prove that this was zoonosis all the way from the start. And then when we actually talk to the scientists, they're some of the people who were most open to this and most concerned about this at the start of the pandemic, which I think a lot of people are actually unaware of and need to be made aware of. So
yeah, I mean, maybe just a couple of comments to that, because I mean, this comes up all the time, right? And it's frankly ludicrous, right? Because I mean, it doesn't make any sense in the first place, right? Is that, of course, there's no big cabal of virologists trying to have some sort of preferred narrative here. And I'll say the criticism leveraged at me early on was always like, oh, but you never took the lab leak seriously. I was like, no, actually, I did take it seriously, and I was quite convinced by it. And people was like, oh, you're just saying that, right? We don't believe you. And then, of course, my email to Tony Fauci comes out, which makes exactly that point, right? So it's like, well, so there you go. As I said, I took this very seriously. I was quite convinced by it, right?
But they haven't then gone on that journey with you. They've just stayed at that point.
Well, but then the problem, then it's the moving goalpost, right? Because then, oh yeah, okay, he took it into, but then how did he change his mind so quickly, right? So surely something must be going on here. And it's ludicrous, right? Because when it comes down to it, and I mentioned this many times, is that it's just a scientific process, right? And when we look at how Mike and I got into this, is that of course we disagreed around the science paper. I told Mike directly that I thought it was too naive. And one thing he mentioned,
the science letter, right?
The science letter, yeah, the science letter, the Bloom et al. Letter, where from my perspective, where it was like, look, the letter does several things. It also says, for example, I mean, it makes it appear as if we literally have no data from the WHO report, for example.
Yes.
Which, from my perspective, was just not true, right? I had noticed, for example, the clustering of the cases, which was in the WHO report. I did a long thread on Twitter around this, where, you know what, if you look at the evolution of just the clustering of cases itself, they cluster right around this particular Huanan market. Also in that report is the fact that if you look at early excess pneumonia death, it's the same thing. Yeah. I didn't do all the formal analyses on this, which is, you know, what Mike has been doing and doing much more carefully, which is super important, right? But these things were in that report. You had to look for them because the report itself doesn't talk about these patterns, right? So you actually have to dig through the data and the visualizations and dig this out yourself, right? Yeah. But that's very important. And importantly too, there was like, look, we looked at all these influenza-like illnesses and we looked at all, and there's just nothing that we see prior to the emergence of the outbreak itself, which was fully consistent, for example, with their genomic data and what the genomic data actually told us. So, and I think that's where the good science comes in, where we disagree on some of these initial points, but we don't disagree on the fact that this should be science-based, this should be focused on evidence, and we should just look to that. Yeah. And that's why this has been such a tremendous collaboration, right? Because we did come into this a little differently, but at the end of the day, we totally agree on the basic
principle. Which is always good. It's always good to have that heterodoxy up front rather than just having the same voices in the room.
Yeah, absolutely.
Can I just add one more thing? I feel like you guys want to move us along, but just on a sort of personal note, it was interesting because that letter was, let's say, not the highest point of our personal and professional relationship.
You're friends now, though, right?
What's that?
You're friends now, though, right? Because I put you in boxes.
We were friends.
We were friends at the time.
Oh, okay. We were friends. We were friends at the time. But Kristian was not happy because he frankly had a clear picture of how that was going to play out, and frankly, at the time, he did have a clear picture of just what he said, you know, how much was in the WHO report compared to how it kind of came across in the letter of like the things that we've talked about.
Do you think, Mike, that the letters had a net negative effect on the data we've been able to get out of China? Has it caused them to be more reticent with data?
Let me just sidestep. Let me sidestep that and just finish what I wanted to say, which was, and both Kristian and I are, I think, are kind of proud of this. If you don't mind me saying that professionally we were like, okay, we're not. This is not the, like, as I said, the high point. But Kristian was like, he sent me an email and the subject heading was compartmentalization. so we're like, we're gonna just like
high decoupling.
Like set aside whatever personal, like maybe I'm kind of pissed off about this, but we're gonna just kind of like move forward. And we moved forward, and it didn't happen like right away in terms of how it's culminated in these papers. But it certainly, you know, we even from that point on, we were like planning a potential trip to China together to try to do some stuff. And so all of that was left open so that when there was this opportunity to come together and do this work, we were able to do that. Whereas in a lot of other cases, it would have been like a flame war and the end of a relationship. And then this scientific work that we did do would never have happened.
I think that's a really important story that like people are just not aware of and need to be more aware of.
That's and just one final point to that too is that like the message around like we should keep an open mind and we should keep investigating. Nobody disagrees on this, right? Yeah. I mean, I said this yesterday in relationship to our two recent papers, that this is not the end of understanding the origin of COVID-19, right? This is the beginning. It gives us the focus that we need, which is that market in the middle of Wuhan, right? But the problem is that there's so many things we still don't know. And of course, we need to figure that out. Right?
And that sort of rubbishes the idea that people are saying now, look, this started with the Lancet letter. People didn't want to investigate it. They wanted to squash it straight away. And this is just carried forward to the modern day where we've now got these papers. It's all definitive. It's all done. Nobody wants to discuss anything. Whereas it's just nonsense, really. When you
it's complete nonsense. I mean, what we don't want to discuss is that all the crazy conspiracies around this is like, oh, you're all the cabal and you're all the Tsunati, you know, it's like, it's not helpful, right? That if and I think again that's what has been so productive between Mike and I coming through this a little differently is that it's fine to disagree, right? I mean, scientists disagree all the time. People in my own lab disagree with me, and turns out they're mostly right. Right? Is that that's fine. It's part of science, right? And in fact, it's part of the scientific method that makes it enjoyable, because it's not about I must always be right. You know, I say this, so that's how it's going to be, right? That's not how science works, right? It really isn't. But when it comes down to these like discussions around a lab leak, for example, is that look, we have actual evidence, and whatever idea you might have around how this virus emerged, it needs to explain that evidence, and it needs to explain it well. And it just doesn't, right? It really just doesn't. What does explain it is the one that we have laid out since the very beginning of this, which is that SARS-2 looks an awful lot like SARS-1. In fact, we had already expected this to happen, and now it happened. Right? Yes. It is perfectly explained by available evidence, right? And a lab leak is just inconsistent with well, the vast majority of evidence.
We're going to come on to that evidence shortly. Philipp, did you have a point to make?
Yeah, just maybe some observations. So I think, you know, Kristian had a bit of a better intuition what social media would do to the narrative of the science letter, because I think
even if it was a good, like high-minded, humble idea.
Exactly. So this is a bit of a problem. This is a bit of a problem that we noticed, is that scientists are unequipped to deal with, you know, messaging on social media, because people are often not aware how scientific processes work and how scientists communicate. And when a scientist, for example, communicates, blah, 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 but there's still this and this uncertainty. But people say, okay, yeah, this person says there's some uncertainty. And then there come the lab leakers and they say 100% it was like this. Everybody else is a shill. And then it's like, okay, there's one person that admits there's uncertainty, the scientist, and then there's a person that seems very confident in what they're saying. So, seems pretty clear to me what's going on here. And this is just, you're losing the fight. And the problem is, no matter what you do in that space, it's not going to work. And Sam and I, we were the same. I was fighting for half a year on social media saying, just because all these MAGA trolls make these conspiracies doesn't mean lab leak shouldn't be taken serious. I talk to people saying, hey, look, I worked in the lab, BSL2 lab. I know, I had people stepping themselves.
We had Yuri
with a
on
virus
to discuss his concerns at the start of the, when we were both quite concerned about it, and we had a nice chat, and there was nothing wrong with that at the start, but then we've moved on from there.
So that's the thing, right? You should not, discard something just because people are crazy online. Doesn't mean you should discard something, but at some point, right, when the uncertainty narrows and the likelihoods get a bit clearer, you should move with the evidence. And this is something that doesn't seem to happen online, because online it's all about, sticking to your guns and sticking to your audience. And if you're a lab leak influencer, you will never change your mind, because your audience doesn't want you to change your mind, and you will just make up stuff. And so the problem is when you then deliver into that hot mix, which was at the time, I think, going already for almost a year, a letter from, prominent scientists saying, hey, we really have to look. And now we have the same problem to a certain extent with the Sago report, where they say, oh, let's look at biosafety again. You just, you're not doing
rather than getting that scientific idea across as being open-minded. It looks like giving 50-50 equivalence, which then gets run away with a life of its own, even though that wasn't the message that was intended.
And I think this is a learning experience that all of us went through. I think the ones that got the most harassment went there the fastest. I think, Kristian, your proximal origins, you're gonna
and frankly, back to the letter, I had this initial, it was March 22nd, March 21st of 21 that I sent an email to Jesse proposing some sort of perspective in science. And then we talked a little bit, and then I kind of was busy with other stuff and kind of didn't do anything. And then a few weeks later, if I recall correctly, there's this draft of a letter that circulates with and I think anyways, it wasn't drafted by me. And it was just a kind of laziness thing that if I had drafted it, I would like to have thought, I would like to think that I might have communicated my own position, which again was, I do not think that this is a very likely scenario, but we need to investigate it. And so that sort of distortion has then carried on right to the present day, even into, as you say, the Sago position of this essentially dishonest assertion that, well, we don't know, so it's 50-50.
Yeah.
Well, what other part of science, do we say there's some uncertainty, therefore both hypotheses or all hypotheses are 50-50? Do we do it with creationism versus evolution?
You reach some actionable level of certainty, and then we move on, because we're never going to get 100%. Do we do it with climate science?
Do we do it with vaccine safety? No. The scientists, particularly, not non-scientists who are just on Twitter, but the scientists who are involved in this, who are repeatedly going back to that refrain of, well, we don't know, so it's 50-50, should be ashamed of themselves. That's the most unscientific thing that you could communicate to the public.
They get very upset with you using the word dispositive, Mike. That is big no. Naughty. That got them all riled up.
All right, so that was a good introduction, I think. So Sam, what's on the list?
So I think we should probably move into the evidence. To be fair, I guess there's kind of three main points that I kind of identified: that the clustering of the early cases, the clustering of the environmental swabs, and then probably the phylogenetics. Mike, do you maybe want to tell us about the main points that make you quite convinced that this virus did come from the Huanan seafood market? What do you think are the main points that people need to be aware of?
Sure. So spatially, again, what the WHO China joint study did was locate or interview the COVID patients who were identified as having, the earliest cases in December of 2019, 174 of them in Hubei province, most of them in Wuhan. And of the 164 within Wuhan, we were able to get, pretty precise coordinates of 155. Okay. And so of those 155, interestingly, the majority were not epidemiologically linked to the market.
So just for people watching, that means that a doctor didn't say that, they were working at the market, or they had a friend who went to the market or something like this.
So let's be really clear here, because there's two kinds of associations here. There's geographical, and then there's epidemiological. Epidemiological is exactly that. You interview the person and you say, did you work at the market? Did you shop at the market in the 2 weeks before you got sick? Did you have contact with someone who worked at the market? Do you live with someone who worked at the market? And so most of the people actually who were identified in the hospital amongst those 155 cases, the answers to those questions were no, 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 and no. And people had taken that and said, well, 2/3 or 3/4 of the people who were identified in this exercise as being the earliest cases in the city, they had no link to the market. So, this didn't come from the market. This was one of the more, leaned upon arguments that the epidemic did not start at the market. Okay, so then there's the geographical association, and so that's what we're focusing on now. Okay, so let's just take all of those cases together, whether they're epidemiologically linked to the market or not, lump them all 155 together and look at where they live, and to do this correctly, you can't actually just look at where they live and say, it looks like they live around the market. Although definitely it does look like they tend to live around the market.
I do need some statistics to really make sure.
What you need to do, and what we did, was have some sort of expectation of, like, how close to the market do they live? How centered around the market do they live compared to what you should expect by chance? And to do that, we used demographic data from Wuhan, from a resource called worldpop.org, which breaks Wuhan into 100 meter by 100 meter cells and tells you, of the 800 square kilometers of the greater Wuhan area, how many people live in each of those cells, which tells you the density of population in different areas. And even better, because COVID doesn't just affect all age groups proportionally. You're more likely to end up in the hospital if you're over 65, for example. We took the age patterns of the 174 cases that were observed in December, and then we transformed the demographic information in Wuhan to match those age groups. And so we're looking at, okay, let's map 155 cases, 155 people in Wuhan of those age groups, let's just sample them one time and see where, you know, a random sample according to the density of that age group in Wuhan would live. So you do it once, and then you do it
Right.
10,000
It's
more
like
times.
bootstrapping
Yeah.
10,000 times to see how it would, when you
Yeah.
pick it, how often would it come up?
Exactly. And then you ask, with the observed data, what's the median distance of the actual cases that we did observe to the market? Is that closer than these 1,000 or 10,000 samplings from the, you know, the actual population of Wuhan? And yeah, it's off the charts. You know, it's the p-value is less than 1 in 1,000 for the distance. So they're much closer, and they're not just closer. They are just ridiculously centered on the market. And we also did something called kernel density estimates, where you essentially sort of work out the probability distribution from which those cases were sampled, and ask, you know, what's the peak area with the highest density of cases? You're not looking at where the market is in this picture. You're saying, of those cases where they're scattered around the city, give me the peak, you know, 1% highest density.
And when you do that, I'm guessing it lands right on top of the market. Or have I given away the punchline?
You've guessed correctly. Okay. And it's just remarkable. And if you understand this, it's in figure 1 of our paper.
They'll all come up, by the way. I'll edit them in so people can see the images.
Excellent.
As you talk through them. Yeah.
Put it right here.
Yeah. There you go. Yeah.
So if you understand that analysis, that it has nothing to do with where the Huanan market is, we're just looking at the location of the cases, that the information flow goes like this. A bunch of people end up in the hospital in late December in a city in China. Some astute doctors at one hospital notice that it seems like an unusual kind of presentation of pneumonia, and they're not infected with any virus or bacterium that we know of. Those same doctors 2 days later realize that more than half of these patients in their hospital work at the Huanan market. Public health officials come in and start asking people, where do you live? And noting down for all of these cases their address. A bunch, several weeks later, an effort is made to collect all of these cases, and we have now address information. That information then goes into our analysis, and 1/3 of a square kilometer out of 8,000 square kilometers ends up with the highest density, and the market is dead center.
Very interesting, and that holds, I believe, for both lineages. It holds for the Chinese data that you talked about, and Weibo data, so social media data as well. They all kind of show the same thing. It holds for epidemiologically linked and not epidemiologically linked as well. However you slice the data, you still get this clustering and centering on the market. Is that correct?
Yeah, and let me just highlight very quickly, because I've been going on forever. Then I'll stop.
We're going to bring Kristian back in a second.
Let me highlight that last point. It's absolutely crucial. The unlinked data. Okay. Now we do this whole exercise where we kick out the 35 cases that they worked at the market, or they shopped there. Kick those ones out and just look at the 120 that had no link to the market. Those ones also, same thing, lived right around the market, centered around the market. Huanan market is dead center in the kernel density estimate. And in fact, they lived significantly closer to the market than the cases who worked at the market. So what's the explanation of that? The only explanation of that makes sense is, well, the people at the market who worked there, they're getting infected at the market. they don't necessarily live right around the market. They live wherever they live in the city. But the people who are not linked to the market are living closer to the market because the virus initially infects like vendors at the market, but then those vendors are going and grabbing a coffee or grabbing a lunch at shops around the market,
interacting with customers who come to that market specifically
community.
and this kind of thing.
And that's how it gets into the city, and that's what we see the snapshot of in December. And then in January and February, it's a completely different picture. The virus is everywhere. It no longer is centered on the market. It's just the highest density of cases is where the highest density of people live.
Yes, and that's actually what the Weibo data shows, right?
Exactly.
Just clear, because the Weibo data is not early enough to look at these February cases, right? That only becomes later in January.
Okay, that's a good.
So that's kind of, just to summarize again from like my perspective, it's super interesting. You have a data sample, you take out all this data that point to the market and say, hey, maybe it's you because maybe you're screwing up the whole data set. I throw you away, and then you look at the data and it's like, fuck, it's still all around in the market. It's like
it's even closer.
Yeah. So this is really the strongest evidence that we can have, I guess.
Just to be clear, I mean, this is what Jon Snow did, right? Cholera. That's how that pump on Broad Street was identified. It's this level of triangulation.
Yeah, exactly.
It's exactly the same.
Kristian, we can't just nod along. So I've got to bring the counterpoints in that people have to this. So there's been some people, and again, our favorite author was posting this today, that some of this WHO localization data is not sufficiently high resolution to support this conclusion. The clustering's low resolution. Some of the age data is not good enough. The cases could still be associated with older populations. What do you make to these comments that this data isn't up to snuff and maybe it's too low resolution?
We built in these are essentially error models, right? I mean, the things you need to be worried about here is like errors in the data points themselves, and then you need to which we can control for, and in fact we do. Right, so we can introduce a jitter, for example, saying that, well, we say it's here, but now let's just introduce a random jitter, and then we do our analyses, and we get the same results. Right, so that we control for that in the analyses, and that's relatively straightforward. I'll say the one thing you have to consider is that is there some sort of special ascertainment bias here, where, well, very specifically they only sample in this particular neighborhood, right? That you might
so a systematic rather than a statistical.
Exactly. Absolutely right. Yes. And that, I'll say that I mean, we look at that closely too, right? And in fact, we also go in and do some analysis and saying, like, what would that ascertainment have to have been before we start losing significance in our results here? But again, that if you look at these early stages, there are limited concerns, no concerns around these types of ascertainment biases driving the signal, right? And what's really important too here is that we're not just talking cases, right? Hospitalizations, it's the same thing, right? Early excess pneumonia death, it's the same thing. And all of these, if there were some sort of systematic biases, would have to have been different, right? And I'll say, especially this idea around the hospitals, is that it's a very clear link back to this particular market, which is also, a lot of people like to bring up other places that are close to the Huanan seafood market.
People talk about the Wuhan CDC, for example.
Yeah, for example, the Wuhan CDC, right? The thing is that there are no epidemiological links there. It's not like people at the Wuhan CDC all of a sudden ended up at the hospitals. And then that's which is how all of this got started, again, to Mike's first point, right, is that this was how this was detected. So they're not workers at the Wuhan CDC, they're workers at the Huanan seafood market.
So there's another thing that we have to kind of, make it easier for our followers to understand. So there's multiple lines of evidence. Now we covered one line of evidence, which is you look at the cases, you look at the hospitalization, and you do this geographic, mapping, and you see, no matter what you do, no matter what biases you consider, it always ends up at the market. And then, of course, we went to the market and we found stuff at the market as well. And there's quite a lot of things that you managed to get out despite, opposition. Let's put it like that.
I think people outside science don't realize how anal, if that's the right word, that scientists are when it comes to errors and potential biases and stuff. That like 90% of any analysis I've ever done has been like error analysis, boring error analysis. Is this what I think I'm seeing? What's the level of error? Be conservative about those errors so that you can be confident about your results. And people don't really see that. They just think, oh, well, if that's the result, I guess they didn't consider this and this. But you've considered so many of these errors.
We keep, and again, it goes back to the first point about how Mike and I started working on this together, where there were points of agreement and points of disagreement, right? Is that we constantly challenge each other, constantly. Like you have a new result, well, you got to have the whole group agreeing that, yeah, okay, that's actually the result and it's meaningful, right? Because we always bring up alternative scenarios, like, but what about if it's this and what about if it's that? Yeah. And much of it gets built into the models, right? Again, the concerns about ascertainment, for example, is a real concern, right? The concerns about noise in the data is a real concern, which is why we address it very specifically.
Well, I remember contacting Mike when he did, and he was busy at the time, but when he did the initial study, which was kind of the lead up to this, you were already considering these ascertainment biases, Mike, with the
science perspective.
Yeah,
I think Mike thought it was all ascertainment bias.
So
right? I mean,
exactly.
I think that's how you got into it.
I wanted to
challenge yourself.
I was totally open to the possibility that it was all ascertainment bias. In fact, I had a message from another scientist who I have the utmost respect for, who said that he had heard that actually the way China set up their pneumonia of unknown etiology system, which is this shield that they set up after the first SARS, so that they could catch people with pneumonia that couldn't be explained by other things and was possibly a new coronavirus or, new avian flu or whatever. That the way they had set up that system was they monitored wildlife markets. Would make some sense, right? That's where these things are going to come from. And so I was like, okay, then it probably is just a bunch of ascertainment bias. And
they've seen this before, so they're hypersensitive to it. They're ready to look for these cases that are linked to the market.
Yeah,
and of course they would catch it first at the market. Like now we know,
because you only find it where you look. Right?
Exactly.
Yeah, I mean, I said as much back in January 2022. I said, like, well, but maybe it was just detected there because that's what they looked, right? But it turns out that's wrong. I mean, that's the key point here. And actually, because as Mike, papers beautifully shows, that it wasn't actually that system that detected it.
Yeah,
that's
the clever
clinician.
The timing was after when that kicked into action later on in the.
It was the doctors in the hospitals.
Yeah, exactly. It was doctors, smart doctors in the hospitals.
Yeah, despite some cheeky publications where the people at the China CDC have taken credit for the PUE system detecting it, which it's not great. It's an important point, and you don't. If that's not how it happened, it's actually really important that people don't think that that's how it happened, precisely because if they think that's how it happened, it then distorts these very fundamental questions like this, that if people think that's how it happened, then of course you're going to think that there's ascertainment bias and that the early cases are tilted toward ones at the market because that's where the street lamp was shining, and that there could have been cases all over Wuhan that were just simply not recorded. And that's the main objection that we've seen from people who are committed to the lab leak theory, that it was just an amplification event, that there was cryptic circulation all over the city and so on and so forth. But no, when you drill down, just one after one, these assertions turn out to be incorrect, and the data tells a different story. If it had been otherwise, I would have published a different perspective in Science, and we would have published a perspective or a couple of papers showing that was a lab.
Yeah.
Good.
I mean, and I think a few important points here, right, is that there because the genomic data has always been inconsistent with this idea that it was widespread by the time is what was detected, right? The genomic data very early on just told us that actually that's not really what's going on, right? But people kept to that idea. It's like, no, it must be that because of the number of cases or whatever, which use wrong parameters, we now know. But what's really important too is that then, and this was in the first WHO report, is that, look, we really need to go back to the blood banks and do a really, good study of all these early samples in October, in November, in December, and see when do we start seeing antibodies to this virus, right? Which was, again, it was proposed in a WHO report. And for the longest, it was like, oh, they're not doing it and they're hiding this data. And it was like, guess what? The data was published just a few months ago. What does it show?
Report or something, wasn't
it show exactly, yeah. What does it show? Well, it shows exactly what you would expect based on the genomic data, which is that no, this virus wasn't widespread until we actually detected it. Right? It's inconsistent. It's in fact, it's disproven by the data, right? When you bring it all together, but it's not a single line of evidence that we're using here, which is a really important, like people love to focus on just one aspect of one particular data.
Sort of divide and conquer what I've seen. Oh, there's a little sliver here and a little sliver here maybe. Yeah, but then you need all these slivers to line up exactly to be able to cut down the whole thing.
And if I could, I would like to just say credit to SAGO and the WHO for facilitating that. My understanding is the 43,000 October to December serum samples being published was facilitated through the SAGO process. And that is what a body like SAGO and the WHO is great for. Yeah. What it's not great for is the specialized independent scientific studies by experts like we are, and they shouldn't be confused. Okay, and this is something that is really important. There are commissions left, right, and center, and you can call yourself a Lancet commission, and it sounds.
It sounds mighty.
Like it has a certain sort of it has the imprimatur of a respected journal, but it doesn't necessarily mean that there's a deep level of expertise for every particular thing that commission might then report on.
Very specifically for that one is very shallow, right? I mean, it's chaired by an economist, first of all. I mean, I think that's deeply problematic, right? This is one of the most complex questions of the pandemic is the emergence of this virus, right?
Yes.
And to think that, I don't pretend to know the physics that Sam, you do, right? I mean, I'm not an expert on everything. I'm pretty narrow, focus on some of these things, which happens to be things like emergence and evolution, right? But if I'm not going to go and saying like, look, now I'm going to chair a committee on why we have inflation right now. And then I'm going to get a bunch of people on that with all kinds of random expertise. And then we're going to make a decision on what we think is the most likely here. It literally makes no sense.
Is that universal inflation or is that economic inflation?
I don't know, right? I mean,
exactly. That's the point.
That's the correct answer.
Which just to be clear here, right? Because I mean, Jeff Sachs, who we are talking about here, he's himself has said that he doesn't know what a furin cleavage site is, right? And it's like, well, if you don't know what a furin cleavage site is, and you base your entire argument on the furin cleavage site being really weird to you, shouldn't that rather tell you that maybe this is not the commission for me?
That's why I get
and
you
maybe
guys
we
on
should
to
have
tell me
somebody with the actual expertise here. I think it's really important, right?
And this is this has become like this sort of rallying cry that you hear over and over again from people again who are committed to the lab leak theory and really don't enjoy when data-driven papers come out that show that the lab leak theory is really not viable. They keep saying, well, we need an independent investigation. Okay, and they're looking to some sort of thing like like Sago, except maybe more populated by people who have preconceived notions that it was a lab leak. Well, there's a mechanism for independent investigations, and it's called the peer-reviewed scientific literature.
You've done it for them, and they still don't like it. So
yeah, but this is a very serious point, that it's not commissions where people come up with some sort of democratic consensus on a bunch of people who have really no expertise saying, well, okay, we think it's this. It's the scientists. It's the scientists who actually drive scientific advances.
That's why, because I mean, the unfortunate aspect of this, right, is that we then see attacks of those scientists. I mean, I'm one of the most attacked scientists out there, right, from all across the spectrum, because I do independent research, right? I'm an independent researcher, but I make it very clear. It's like, look, I stick to data, right? And this is what our data shows us. But that's the problem here, is like when you can't actually engage with the science, the data, the evidence, because it really tells you a different story here. Then it comes to like, well, then let's go in and attack the scientists instead. And we have seen this over and over and over again. There's people spending all day on Twitter just talking about that, right? And they
want to talk about anything but the data, conflicts of interest,
because the data inconveniently
things that didn't get funded and these kind of things. The data is just whatever.
Yeah. And I think, we have to be careful that, I'll say, things get overinterpreted in both ways, right? Where, for example, there was a recent study where one of the banal viruses was passaged for 6 times, right? And it didn't gain a furin cleavage site. And some people, took that to mean that, oh yeah, so see, these viruses can't gain furin cleavage sites. And others took that to mean that, oh yeah, this proves that they don't gain furin cleavage site during passage. And the problem is that a negative result like this really, at the end of the day, doesn't tell us anything at all.
Yes.
Right? It doesn't, because maybe it would have gained it at the 7th passage, maybe.
Yeah, they didn't have a control, right?
But it's irrelevant, right? If we had actually seen it, right? If we had very specifically seen that virus, for example, gain a furin cleavage site, that would have told us something very important. That would be a positive result.
I was just excited to see which way they would go. Either admit that, a limited experiment didn't make a furin cleavage site, or call for the GOF to show that it could. Like they were sort of sweating over which one to do. They weren't really. They were
and I think again, because this is where we have to be careful that both camps, if you want here, sort of overinterpret the data that's out there, right? Because this is a perfect example where actually the first study finding the banal viruses in the first place, that's the really key study, right? Because it tells us, what, these viruses are actually very diverse and they're highly abundant and they're all over the place. Right?
Yes.
And actually, they have this, receptor binding domain, for example, that we find in SARS-2 as well. But now we found it in bats, not just in those we found in pangolin coronaviruses, right? These are really key insights that little by little, allows us to sort of peel away of the evolutionary history, the deeper evolutionary history of these viruses, right? And I think it's important, again, that when we talk about what do we think is most likely scenarios? What do we think? You got to take all the evidence, right? You can't just say like, look, I'm only going to look at this. I'm going to ignore all of that because either I don't know how to look at it or I don't want to look at it. I haven't looked at it yet. You can't do that, right? You need to take everything that you possibly have. And then the speculations you just have to put aside, right? We're not talking about, well, but they could possibly have done that, or maybe they could have done. It's like, sure, yeah, maybe they could. But what evidence do we have to suggest that they actually did? And every time we look for that evidence, we come up empty. I think the 90-day, intelligence investigation here in the United States is a perfect example of that. That is not geared towards finding, for example, infected raccoon dogs at the Huanan seafood market, right? It might have been geared to detect some early emails,
secret cables between the ambassador,
like, spook kind of stuff that we can't do as scientists, right? So that is very specifically geared towards, is there actually any evidence for a lab leak here, right? And they come up with the same conclusion that we do as scientists, which is that it doesn't matter where we look for this, we just don't find it. What we do find is actual evidence that points right to a market at the Huanan seafood market.
I'm going to have to bring it on to the second point on that market evidence then. So
which is inside the market.
Yeah.
Right.
So inside the market, the swabs, the environmental swabs seem to cluster on the west side. So Kristian, do you want to tell us about why that's important so people understand?
Yeah, so I'll say that this is so all of this is actually based on a report that was released and quite heavily reported on in Chinese news media back in January of 2020, where the Wuhan CDC and China CDC went in and sampled the market, environmental samples, I believe, on the 1st of January and the 12th of January, and they had about 585 samples or so. And all of this was reported in a report, and I can't remember exactly the numbers, but I can't remember the numbers, but quite a lot of these were positive.
79, right? I think.
So 29?
79.
Yeah,
almost 80.
I think it's 70. Yeah, exactly. So they actually report on all of this, right? It's in this report, which is not a public report, but importantly, this then get reported on in official Chinese news media. And it says the positivity rates. They say that, look, it all clusters, yes, in the western side of the market, but also really importantly, where we now know that they sold animals, right? Which is like, holy crap, this is actually January 2020 we're talking about here. This wasn't really widely reported outside China, and importantly, that report was not actually made publicly available at the time. That then gets leaked to various news outlets later in the year, I think May or June of 2020, and specifically we got this via the Epoch Times. And what is interesting is that when I look back to our, scientific discussions around this, we actually noticed it at the time. We even noticed sort of like, oh, there's this like sort of clustering or whatever. And then came, I think it was Delta, right? So actually this must have been 2021. So then came Delta, and then we got way too busy with other things. So we never looked at it again, right? And then, at the time where we're starting to talk to Mike about other investigations and stuff like that, there's this, Twitter user, Baba, had published this amazing map.
This is great, yeah.
Yeah, it's unbelievable, right? I mean, this is why we're talking about actual sleuthing that just blows my mind, right? Baba had made this map where he had, taken all the different photos from all kinds of resources, and he had plotted it onto this online map and put it all in there, put where were the positive cases and stuff like that. And I was looking at that, and I was like, wait a minute, didn't we actually have some more data on this that we looked at like a year ago? Let me just go back to that. And then I looked back at it and was like, how can I have missed this? All the details in there. What stalls were positive, what stalls were negative, where they were, how many were positive, like all these stuff was right there. So I started looking at that with my more simple analyses, right? I was like, holy smokes, this cluster right where we have, these animals being sold, which they had reported on in 2020, right? January, I just missed it. But then really importantly, it was like, this is the shop that Eddie visited, Eddie Holmes, in 2014, right? All this positivity stuff is like right on top of this shop that Eddie visited at the Huanan seafood market in 2014. And guess what? Took photos of raccoon dogs sitting on top of birds, right? So
I think you had fines and stuff and notices after that showing that the animals were still being sold up to the
so that comes additionally. So what's important is, first of all, like, we then know what objects are we actually talking about.
Yeah, this is super.
We're talking about here where we have this specific clustering of where in fact we find the vast majority of positivity is that, which is this, this particular shop that we're talking about here, is that it's things like cages. Yeah. Right. So a cage is positive. And Eddie was like, yeah, that's, I took photos of these cages. And then it was things like carts. And he was like, yeah, if you look at the back of my photo, you can see like 2 carts, and 2 carts were positive, right? 2 carts in Eddie's photo as well. Yeah. And then there was this feather remover, which in Chinese, this can mean many things. And all of this was professionally translated, right? But I was like, so what's with this feather remover? And Eddie was like, zoom into my photo and look at those raccoon dogs. What are the raccoon dogs sitting on top? And they're sitting on top of birds, right? So it's like, you have got to be kidding me. like this, and of course, all of this is 2014, right? We have everything that we basically see as positivity in this one shop where we have most of the positive samples, right? I actually in Eddie's photos.
Yeah.
But then
also the fridge and the sewage, right? Was also
well, and then we
positive.
know from later studies too that we actually know that the sewage drain system outside this shop, which in fact these cages sit on top, right? We also know from the Gao preprint, we know that's actually positive as well. So that's important. But of course, Eddie's photos are from 2014, right? It's like, yes, but were they actually selling animals in November and December, for example, of 2019? And that's where Xiao's study is really important, because he had reported that, look, they do this across market, but we didn't know the details of all the different animals being sold. That's what we then report on in this paper, right? Where we report on that, yes, several susceptible animals were in fact sold in November, for example, raccoon dogs, but there's also badgers, which were also positive, for example, with SARS-1. The same is true for raccoon dogs, right? And other animals that we assume to be, that we either know or assume to be susceptible to infection with SARS-2, all on sale, all the way up to just the start of the pandemic, right? So yes, that gets reported here too. So when you take all of this evidence too, is that the clustering that we see here is not actually just associated with the cases and hospitalizations and excess deaths. You go inside that market, it also very specifically clusters where they sell these kinds of animals. And you can say that, well, that's a funny coincidence if somebody walked in with the virus and then taking it from there, right?
Yeah.
It doesn't really make sense when you really have to explain the evidence. And of course, there's the PCAR study showing the likelihood of multiple introductions, right?
We're going to come on to that.
Yeah,
Mike, the counterpoint to this piece of evidence that people always bring up is, but what animal? You don't have an SC2 positive animal that you can point to, and people get, they just say this over and over again like a mantra. How do you respond to that pushback that we don't have an SC2 positive animal to actually look at? Because the Chinese samples, they say, look, we had no positive animals when we looked at this on the 1st of January.
You're muted, Mike.
Mike, I absolutely agree that we don't. But there's some reasons that we don't, and there's some important comparisons and contrasts to be made. So back to SARS-1. SARS-1, first cases 2002, cases through 2004, no markets shut down, no farms shut down, samples eventually, after months, were taken from animals in markets, and that link was made. But this market was shut down within less than a week from when the first cases were really, crystallized in people's minds as connected to a new outbreak. Totally different scenario, okay? And so no relevant live animals were sampled as far as we know from the market. They did go in and sample some live animals, but they were things like cats wandering around,
stray cats,
mice that they were able to sample, a few things like salamanders that were still on sale. But the raccoon dogs, the foxes, the badgers, the marmots, the Sika deer, all of these things that you can read in table 1 that we report were present at the market, certainly in November. And we've got also reports from CNN with photograph or video evidence that are pretty reliably dated to December of these same animals. So they were there, but they weren't sampled for whatever reason. And so this idea, and it's really kind of pathetic that you hear that,
and you hear it a lot.
You hear it a lot. We don't have a positive animal sample. And you also hear, and 80,000 animals have been sampled. Well, not a single one of those was one of the species that is, relevant and was sampled live at the market. Not a single one.
Mm. Maybe I interject very quickly. So there's different explanations, and some of these explanations are, the Chinese were covering up the animals. Some people are saying, this is they had an advanced warning or something. But I think what is the most likely explanation, also when you look at the reporting from some people at the time, it was like, people,
the traders just scarfed
the humans there, like the humans thought, shit, it's SARS-1 all over again. And so they got rid of the animals as quickly as they could, because of course they all were panicked that, oh fuck, we're gonna be blamed. They knew SARS-1 was from wild animals. So everybody who had the wild animals thought, oh shit, before the authorities come, I'm gonna get rid of my animals. I'm gonna ship them, I'm gonna sell them, whatever. I have to get rid of them. So this would be a non-conspiratorial explanation why the animals are gone, because some people say, maybe the Chinese are covering up that these animals were, or they're faking false positives. So no, that's not the case.
So it's hard to know, but there's reasonable other explanations as to why we might not have.
Yeah, I think that's important, because we should refrain from speculation on this, right? I think what the key point is that the animals were actually there, right?
Exactly.
They were for sale, leading all the way up to the pandemic, and then we know based on reporting that none of them were tested.
People also like to point out that the human cases were more diffuse compared to the West side, but is that something we'd expect when humans walk around and move around and we miss a lot of the cases? So tables don't move around, caged animals don't move around, but humans, do.
Yeah, so humans move around, and I think that's, so again, the human cases do cluster to the Western side of the market, right? But much strongly, they don't really cluster where we necessarily find the animals, although some of the early cases are right adjacent to and stuff like that, right? Generally speaking, the human cases are clearly more diffuse, which is no surprise, right? And we have to remember that most human infections are asymptomatic, so people probably don't notice. We have heard, there's been stories like early on about being infected, sharing bathrooms, for example, and like Mojang, what is it called, like parlor type of stuff. And we actually know where these locations are now, thanks to really incredible sleuthing from Alex, Christoph on the paper, right? It's not in the paper, but we know that the places we're talking about here are actually right where these animals are being sold to. Very specifically, I should add, is on top of this one shop we're talking about.
So wait, can I repeat
is actually right. It's right there, right? And the parlor.
You're saying where the animals are, and then the bathroom for the humans is right on top.
Yeah, well, the bathroom is right adjacent to that shop, and the parlor.
And the mahjong is on top. All right, great.
It's right on top. Yeah. So it's not in the paper, right? Because it's a little too much sleuthing here, but we'll probably talk about that at some other point. But it all sort of makes sense, right? Why would you see these things? Because having more diffuse human cases, but they are over in the Western market, and the earlier ones are right adjacent. All these things, that's exactly what you would expect, right? It really is exactly what you would expect. I also want to say, just to take it back to not having an infected animal here in SARS-1, is that what's really important to remember too is that the vast majority of animals, including civets and raccoon dogs, tested for SARS-1 in places in which you had outbreaks were also negative, right? It's just that many more animals were sold and many more animals were tested back then because they were specifically tested, but the vast majority of them were in fact negative. So the likelihood of finding a positive animal in itself is very, very low, unfortunately. And of course, if you don't do it in the kind of animals you actually need to do it in, well, then it's always going to be zero. Right? It's not going to move from there.
The third big piece of evidence, Michael, come to you, is this, which is probably a little bit harder for people to understand, is kind of the family tree of these viruses, the phylogenetics that suggests that this virus is introduced to humans at least twice. Can you tell us a little bit about that and why that's important?
Yeah, absolutely. This actually goes back to work that I did with Jonathan Picard and Joel Wertheim, trying to work out the emergence of the virus in Europe and North America. And so let's just focus on the North America side really quickly. So there was the first COVID case in North America was a guy who had traveled back from the Seattle area to see his family in Wuhan, arrived back at SeaTac Airport January 15th of 2020, was diagnosed, isolated. His genome was sequenced, first genome in North America to be sequenced. And then about 6 weeks later, Trevor Bedford and others at the Seattle Flu Project started sequencing samples from the area and found a person who with no travel history to China who had the virus, and lo and behold, it was only a couple of mutations away from this January 15th patient. And then more and more were sampled, and they were similarly a couple of mutations away. And that's not very many mutations. And so the immediate inference was, it looks like this guy on the 15th of January actually his virus escaped isolation and had been cryptically spreading in the community. We generated some technology to put that to the test and actually simulate epidemics starting with that genome on the 15th of January, and then asking, actually, would you see this clean break of 2 mutations between that virus and everything else without seeing, for example, transitional forms that are 1 mutation different and so forth? The answer was a solid no, surprisingly. So even 2 mutations can tell you a lot. And it's really the same pattern that we see early on in the outbreak here, where we have these 2 lineages, lineage A and lineage B. They differ by 2 mutations. And what we see when you put together all of the genomic evidence is that there's something like a 99.5% probability that this 2 mutation difference actually does indicate two separate jumps for these 2 lineages. And that, interestingly, the lineage that's actually further evolved away from the bat viruses, lineage B, probably, not necessarily, but it looks like it's the one that jumped into humans first, and that lineage A actually jumped later. And if you kind of force the scenario that most people who think that there was a lab leak favor, which is that there was a single introduction and that it was the bat-like lineage A that was at the root of that introduction. Actually, what we show, and this is new in the paper compared to the preprint, is that actually kind of breaks the molecular clock, that lineage A at the root of a single introduction actually is not consistent with a molecular clock. And so put the 2 papers together now, and we haven't touched on this yet, and I'm glad we have a chance to get around to it, because one of the other really major things that we found in the other paper was that although lineage B had been the only lineage associated with the Huanan market, the human cases and the environmental cases up until the point we were finishing these papers had all been lineage B at the Huanan market. What we found was that we had a couple of lineage A cases where we had location information, and they were both near the market. And just sort of broadly speaking, they were so near the market that you just couldn't explain that by, again, the population density patterns of this virus. And lineage A actually had a slightly stronger association with the market, even though it had never been found at the market. the cases that had no link to the market were so close to the market that it actually looked like an even stronger association. And even though there are only a couple of them, it's kind of like me saying, I'm going to shuffle this deck twice, and each time I'm going to pull out an ace of spades. It's really unlikely. It's unlikely to do it once. It's ridiculously unlikely to do it twice. And so that's where we were with lineage A. And that's important because it destroys the idea that the lineage that the Huanan market was just simply a super spreader event, which again is one of the refrains that you hear. Just an amplification event. The virus was emerged somewhere else. Well, no, you don't get a super spreader event and have two different lineages associated with that. A super spreader event amplifies one lineage. And so you put the two together, strong evidence now, and in the paper versus the preprint, Andy McGee and Mark Suchard, who are co-authors on the PCAR paper, brilliant additions to the new paper that allowed us to synthesize a bunch of different evidence into like a single Bayes factor, where you can just say, let's compare the probability of 2 jumps versus 1 jump. That leaves us with strong evidence of 2 jumps. The geographical association leaves us with clear evidence that both lineages were tied to the market. That tells us that 2 jumps happened at that market.
So you've got all these lines of evidence converging in the same place.
Yeah. Also, the model accounts for the case numbers, right, and how they would go, and they could actually show that there is at least 2 introductions, right? Because this is something that I found super fascinating. The idea that, how likely it is, and this is another paper from PK, and I think you were also involved, maybe, Michael, where you basically showed that, look, if a virus like SARS-CoV-2, the original one, it wasn't that crazily infectious. It was very infectious, but it wasn't, it was like a Delta variant. And right. if that would jump over, in some village somewhere, it might not find enough humans to, start a pandemic, to initiate stable transmission chains. And even in a city like Wuhan, with a certain density of people, the odds are like 70% odds, I think was the number, that it would just die out. It would infect, it jumped once, put into a human, the human gets sick, but the virus wasn't super infectious, so it might not just infect somebody else and you wouldn't get it. So the idea is always that there must be a stable interface, a human and animal reservoir interface, or a person that is constantly there and sick, that would jump over and repeatedly, because the first one might die out, the second one die out, but if you have like 10 jumps, 2 of them will make it, and this is probably also what we are observing.
I'm just aware of your guys' time. How are you? Because I could talk about this for another 2 hours.
8 minutes.
Right, you got another 8 minutes. Right, let's finish off this.
Just to put a number on that, to get at least two introductions of lineage A and lineage B, the sort of average number of total jumps, of which some go extinct, that we estimate was 8 at the market.
So the counterpoints that get put to this, Kristian, from people who, again, they do the divide and conquer thing, they say, look, Picard picked certain genomes and kicked them out, these transitional genomes, unfairly. Kumar gets different results. How do we respond to those kind of criticisms that...
Well, so we don't randomly kick out genomes, just to be clear, right?
This is the refrain you hear, though. Like,
sure. And in fact, that's the problem with the Kumar study, is that they're not careful with the data set, and then the model itself is actually inappropriate too. So there's several. There's a data issue, and then there's an analysis issue. The Kumar, they're not set up to actually calculate these. That particular algorithm is set up to create big trees, not actually to accurately estimate TMRCAs, which is.
And I don't think to just be fair to Kumar, I don't think he's weighed in on this matter. I think his analysis has been used by other people to say so.
No, I don't think so. And I think, well, I mean, there are some stronger statements in the paper, I would say, which I just disagree with. Again, based on data, but also based on the model itself, which I find
we're also not naive, so we also talked to some other people that understand it, and compared Kumar to Bekkar, and we didn't ask Jonathan about it. We asked, for example, Spiros to explain to us, why Kumar comes to a different conclusion.
Just jumping in, Kristian, because I have some knowledge of this. So in the Kumar paper, as you say, it's a method for doing stuff with zillions of genomes, which and the clock dating that they use to get a really early date, he actually just kind of steals our clock rate from PCAR.
It's fixed, right?
PCAR 2021. It's fixed. It's not from the data set that they actually are using. It's from our data set, which is inappropriate.
The outgroup is this ProCov2 or something as well, isn't it? I can't remember.
Well, that's a separate issue.
That's a separate issue, which we also actually deal with in PCAR and show that ProCov2 is actually not an appropriate. It's ruled out as the route as would be the ones that they propose in this paper, but it's a separate issue. Anyways, the molecular clock work is sort of a back of the envelope thing using our rate from a different paper, and it's just not a reliable way to
and I'll say, just to be clear, is that it's not a criticism of that work, right? Because I actually do think that what they developed there is quite useful for, again, the stuff
for what it is
before, which is large phylogenies, right? It's not useful for trying to get an accurate date on, for example, the TMRCA here. And really importantly too is that there are the issues around the data. And the reason why we actually need to take out some of the genomes here is specifically because they're faulty. Right. And we know this. I mean, some of these we actually got to check the data itself. And yeah, indeed, they were down to bioinformatics errors. And it's because Mike noticed, for example, that, look, they have all kinds of other mutations that just phylogenetically just don't make any sense at all. So it looks like there's something wrong with these genomes. And indeed, when we look at it, it's like, oh, yeah, it's just a bioinformatics error, for example. And that's not, again, that's not even criticism of that work either, right? Because this is what you should expect. You can't expect that here's a genome that we found on a public database and it's going to be.
No, and I think a lot of the scientists that were contacted said, oh, yeah, we made a mistake. Sorry. Yeah. Okay, fine.
Yeah. They didn't say sorry. They joined us as co-authors.
Yeah, that's even better.
There's nothing because there really is nothing to be sorry about. But this is also why, like, again, Mike has been doing this for 25 years. I've been doing this for 10 years, right? And that's where the expertise is actually important. That you can't just all of a sudden saying like, oh, now I'm going to phylogenetics expert, so now I can totally figure out these studies and criticize these studies, right? Because when you looked at this, and importantly from my perspective, we have generated, I think my lab now is more than 60,000 genomes of SARS-2, right? But prior to that, for all kinds of other viruses too, Mike has pieced together like the oldest HIV that you have, that, there's barely anything left in those samples yet, genomes to generate it in Mike's lab, right? And it's really important to understand when you do all of these things, you need to be aware of the little mistakes and the little errors that come into it, and make sure that your analyses are robust to that, or at least take it into consideration. And that's the reason why some of the genomes had to be, taken out of these analyses. I want to make it very clear, though, that one important analysis we do here is saying that, well, if you look at the genomes just at the Huanan market, for example, and do estimates and when do the timing here, or we do stuff in Wuhan, or we do stuff outside Wuhan, so none of the Wuhan genomes here, we all come up with the same results, right? The TMRCA here and the difference in frequency between lineage A and lineage B, for example, stay consistent no matter how you look at it.
So you've got many lines of evidence that are all converging on the same place.
It all converge to the same thing.
Even if people want to undermine one piece a little bit, you've got to consider all of it if you have an alternative hypothesis, which has to explain all of this. And there isn't an alternative.
And I think that's when it comes to, like, for example, the rooting of the tree, right, which some people have made mistakes around this, saying like, well, the one that's closest to the bat virus, that's got to be the root, right, which is not true, first of all. It could be, but we don't really know for certain, right? So when you do all these kinds of analyses, you need to take, look, we have a molecular club that gives us some information. We have, outline of outgroups like the bat coronaviruses or the pangolin coronaviruses. That gives us some information. And then we have the timing of all of this also gives us information. You take all of that into your model, needs to consider all this evidence. You can't just say, well, I'm only going to do this, or I'm only going to do that. You got to do all of this at the same time. And then if you do that correctly, what you end up with this model that, to explain the body of evidence here, really, it's much more likely that you have multiple spillovers, right? Much more likely. If you don't have that, if you're not willing to accept that, then, well, parts of your model becomes very unlikely, right? And specifically inconsistent with the data, I should say.
Makes sense. Mike, I know you've got to run. Okay,
I could stay on a little longer.
You can stay on a little
longer.
That's
nice. Then we can talk about the conspiracies.
Hi guys. Thanks a lot. I think we're going to talk about some talking great
points. Yeah, Mike, it was a great pleasure. Thank you very much for taking the time. We really appreciate it.
Thank you, Mike. Guys.
And congratulations for the
paper.
Nice publications.
Yeah.
Oh, thanks.
So, Kristian, I wanted to come on to a couple of sort of talking points that people have. So, the first one is that the Huanan market is just a super spreader event. We've seen Jamie Metzel, we've seen other people saying this even yesterday. It's a common refrain. What are the main pieces of evidence here that we can say, look, this isn't just a super spreader event. This isn't a human to human amplification event. Why can we be quite confident that that's not the case?
Yeah, I mean, I'll say, there's multiple lines of evidence. One piece of evidence, for example, is just the epicurve from within the market, which is actually in the WHO report. If you reconstruct saying like, here are the number of cases we detect in the market over time, which is just a standard epicurve, you reconstruct that, what you see is that you end up with a doubling rate from within the market in sort of like the 3 to 4 days, which is fully consistent with what we then later see outside the market. So that's, that in itself is like, so it's not just a super spreading event. Then you can come to this like, oh, but maybe it was an amplifying event, or maybe it was like, well, maybe it was an amplifying event. But then you have the issue again, like Mike was talking about, but you have lineage A and you have lineage B, and they grow at about the same rate, both inside the market and outside the market. So it's, well, I should say primarily outside the market, but whether they're associated with or not, right, is that the growth of these lineages over time is actually very consistent early during the pandemic here. And really importantly too, is that based on our analyses, we were like, look, lineage A must have been at that market because the analyses really strongly suggested, but we didn't know for certain. Of course, George Gao from the China CDC comes out with this preprint and saying like, oh yeah, lineage A was actually at the market. We detected in this environmental sample over here, right? So again, you put all this evidence together and saying like, no, actually, there's nothing that suggests just based on that data that this was simply a super spreading event. Then you add another analysis, which we include in.
I think it's a new one in the update.
It's a new. I can't remember whether we had it as a supplementary figure or whether, but now it's a main figure, right? Where we're saying like, look, we have this social media check-ins, and this was Christoph again.
It's a really nice bit of sleuthing again. This
and Alex is an amazing sleuther, I should say. I mean, it's just incredible, right? I really enjoyed working with Alex, I should say. So
I don't want to upset him because he'll probably like find out all my secrets and my bank details and.
Yeah, no, I hope Alex. It's anyway. We've had some late night sleuthing too, and the guy's amazing. But what's interesting here is just like, well, let's just ask this question and saying, okay, let's just accept that it was a super spreading event. Right? It was simply just a place in which the virus came in. Let's just forget that we think it came in twice, right? Because we're gonna forget about that too. And we're gonna forget about all that data that actually tells us otherwise. Let's just accept like, no, actually, I think it was a super spreading event. Well, then we can ask the question, how likely was it that a place like the Huanan seafood market would end up as a super spreading venue, as opposed to all kinds of other shopping venues, concert venues,
stadium,
whatever,
a concert, a church,
exactly,
whatever.
all the other places you have in which a lot of people congregate. Well, we can do those estimates based on social media check-ins and social media data. And what we see here is the traffic at the Huanan seafood market is very, very low compared to many other venues, including other shopping venues, I should say, across Wuhan. So even just that a priori, where we're saying like, how likely is a place like the Huanan seafood market to be a super spreading event given no other evidence? It's low, right? Very low, in fact. So again, you put all these things together and saying like, no, guys, I'm sorry, but it wasn't just simply a venue in which this virus amplified itself. While we can't disprove it, there has to be clear, right? Proof is, Sam, you're very familiar with proof, right? Coming from physics and math, is that in biology,
I hated proof lessons, by the way, but
that's fine. Yeah, aware, and I remember it from my own time. There, it's like it's not, that's not really how this works, right? This works by you keep looking at data, you look at multiple lines of evidence, you bring it all together and say, like, what does this actually tell us? And it tells us something very, very, very clear. I would say much clearer than we typically have in terms of this, and that points right to that market where all the early hospitalizations and all the early cases clustered around, all the evidence points to that.
So you've got the A and B both clustered and centered on this exact place. I mean, if it was a lab leak, what are we saying? 2 spillovers? It makes no sense.
That's impossible. I'll say if the issue with the Picard study, right, is it's a statistical statement. It's not a proof. If you could actually prove beyond any reasonable doubt that the virus spilled over twice, and they were associated with the Huanan seafood market, the lab leak is fully off the table. It is disproven, right? Fully off the table. The reason why it's not, disproven, I just find it not plausible, right? Which doesn't mean it's not possible. It stays within the realm of possibility, which is why we have to keep an open mind, as any scientist always should, right? But it's just not plausible because of the body of evidence that we do have. And the body of evidence, importantly, that we don't.
And why would this place, this unremarkable place in Wuhan,
of all places,
just so happen to be right at the center of this, looking exactly like SARS-1 did in the past? It's just that idea that they have of
not exactly, right? But all the key components are exactly the same. And one thing we haven't mentioned is just the timing. Right. If we look at the timing here, SARS-1 spilled over in November 2002. That was the first spillover on that. Well, based on all the evidence, when did SARS-2 spill over? November of 2019. Right. That's not, maybe that's just a coincidence. It totally could be, right? But maybe it's not, because you have the way in which bats roost and procreate and all these things which you don't know anything about. Is that these winter months, and also people getting close together, right, is some of the highest risk for not just the virus trying to spill over into the human population, but actually successfully doing so. And I think this is an aspect of this that many people don't have a good understanding of, which is that these types of viruses that are highly abundant and they're highly diverse, they try to spill over into humans constantly, right? Constantly. And many times they successfully do so. But here's the thing, they go extinct. Maybe they can cause a little cluster of cases. Maybe they cause one infection, but no illness. Maybe they cause 2 infections and 1 illness. We never detect these, right? Because they never get onto our radar until, of course, they do. And they only do when you have everything lined up, just exactly right. And to compare that, for example, is that, again, in this lab leak idea, of which there are multiple different versions of this, some of which are entirely inconsistent with each other. For example, the Wuhan CDC, I love that, because it's like, well, but then you can't have anything about the gain of function research, you can't have all of this, right? It all goes away. But anyway, you take this idea about the lab leak is that, look, what we're saying is not that it's hugely unlikely that somebody would infect themselves in the lab, because that actually happens, right? It's a lot rarer than people being infected out in the wild, but it happens, right, at some appreciable frequency. That's not what's so unlikely here. That virus needs to be in that lab before it can spill out of that lab.
Yes.
And that's the part that actually, you know what? the likelihood of a researcher going out there randomly, swapping a butt of a bat or something like that and taking that back to the lab, magically actually being able to grow it, which is exceptionally difficult, right? We have tried to do it, not with coronaviruses, but other bad viruses. Most people just can't do this, including the VIB, I should say. They have largely been unsuccessful at growing viruses, right? You take all of these, they all need to happen upstream for this to spill out of a lab. And then, of course, downstream, well, then that's when you come into, and they need to be lying about it too, right? And that adds another level of complexity, which when you look at this body of evidence, actual evidence, I'm not talking about what maybe they did this or maybe they thought, no. I'm just talking about stuff we can look at, stuff we have, multiple lines of different evidence. They all point to the same thing.
And it's interesting, isn't it? That this was the first idea, was that, why would it just so happen to be Wuhan, or just where this lab is, and so on. It was this idea of sort of parsimony. But now there's so much evidence on the other side that parsimony actually sort of works against them now, that you would have to hit all these things just exactly right. That, yes, I mean, in some freak, 1 in a million possibility, okay, maybe, but we've reached actionable certainty now that this is a very, very unlikely thing to have happened.
Again, I mean, things like parsimony and Bayesian and like all these things that are technical concepts, right, that scientists know well. I mean, most of our analyses, for example, are Bayesian analyses, right? Yes. Mark Suchard, who's one of the lead authors on this paper, is developed most of the framework around this for phylogenetics, right? So did Andrew Rambo, for that matter, and Eddie Holmes. I can keep on, right? Yeah. So it's not like people love to bring up these words. I will say, in the absence of any evidence whatsoever, things like parsimony can actually be quite useful, right? And from a parsimonious standpoint, while the natural emergence of this is way more likely than the lab leak, but if you just look at it like, let me just quickly look at it, because of the Wuhan Institute of Virology, because of the work that they were doing, well, that's why you now have to actually consider this as a real possibility, because some of the ingredients were certainly there, right? So you need to consider that. But parsimony, when you actually have evidence, right? I mean, even again, just to be clear, from a parsimony perspective, natural emergence is still way more likely. So I'm happy to just take a parsimony standpoint on this, right? But forget about parsimony, because we have actual evidence here. And what needs to be clear about this evidence is that you need to consider all the evidence, first of all. You need to be able to, your hypothesis, certainly if you want it to be a theory, but even your hypothesis needs to be able to explain all that evidence. You can't just, this over here or whatever, right? And then you also need to realize that none of this data is ever going to be perfect. I see this coming up frequently from some of my colleagues, right? That say like, I just don't understand. We don't have perfect evidence or perfect data on early cases in Wuhan. I was like, how many times have you tried to actually generate said evidence? Which is an important question. I can tell you that, for example, spending time in West Africa during the early stages of the Ebola outbreak, as well as the late stages too, right, is that actually getting this level of evidence is not easy. Right? It might seem easy if you're a desktop jockey sitting somewhere in the United States and saying like, why can't they get this perfect evidence, right? Is that getting that evidence in itself is actually incredibly difficult. And that needs to be clear, right? What also needs to be clear is that the evidence we actually do have is incredibly detailed. And if you don't believe my statement here, then just think about, if we look at the early emergence of SARS-2, for example, in the United States, what evidence do we have exactly on early cases here? It's poor, right? Yeah. In fact, we have no evidence because we weren't testing, right? So the evidence that we actually do have, while it's not perfect, it's not terrible, and it's certainly a lot better than you could typically. The fact that we just have things like where they actually lived on earlier cases, like, are you kidding me? I mean, this is highly detailed data, right? So it has to be clear that both on one hand, it's not perfect. On the other hand, yes, we all want more and better data, But C, really importantly, is that, but that doesn't mean that we can't say anything at all or make any conclusions based on the evidence that we actually
the only thing that happens is that your error bars get a bit bigger, but it doesn't really matter if
Exactly.
you then have multiple lines of evidence and each error bar is like very small or, you know, a bit bigger.
Exactly.
It doesn't matter. And this is also why I said at some point, after I looked at all the evidence there is and wrote this very long article that probably nobody really read.
That's great. No, it's great.
It doesn't matter. You can assume, you can grant the cover-ups. You can say, look, I grant you 100% leakage rate in the width. Doesn't move the needle on zoonosis. It doesn't. It just doesn't account for the evidence. Even if every virus they ever had would have leaked out of the lab, it still wouldn't move the needle because all the evidence points in the other direction. And the same is true for other cover-ups. You know, assume, let's say, you know, they had the virus. From all we can tell from the genomic data is that this was a pandemic prone virus before it ever saw the inside of a lab. We know this from the mosaic genome. We know this from how it was shaped. We know this from the RPDs that were found in the Lao sequences. Genetically, this was a virus ready to go before it ever saw, if it ever saw human.
And we still see, I mean, just to be clear, we still see that, right? Because when we now find ourselves in the pandemic, this virus is continually spilling over into all kinds of animal species.
Yeah, that's another point.
Right?
Yeah.
And not just that, but in fact, establishing itself, like white-tailed deer here in the United States, for example, and Canada. Are you kidding me? Right? Is so there's none of this, which is sort of like people like, you know, oh, that's really mysterious. There must be some way of like really mysterious explanation to this. And it's like, no, it's unusual, which is true because pandemics are usual. But pandemics only happen when you have all these things come in at the same time.
And people only care when there's a pandemic. They don't.
Of course, because you. I mean, again, I've mentioned this several times. It's like if you're afraid of gain of function research in labs, I'm like, think about the gain of function research that the nature is doing on a daily basis all over the place. Right?
Exactly.
And again, it's trying all the time, right? And only rarely are they actually successful. And just a couple of points to this, because Joel Wertheim, co-author on, you know, leader of the Picard preprint paper, I'm calling preprint. It's not. It's a paper now. Is he keeps bringing up this Everest analogy, right? Mm-hmm. Which is that for the longest, in fact, for all of human history, Everest was there and we never climbed it, right? It was just sitting there. Then we tried to climb it and we were pretty unsuccessful for quite some time. And then one day, 2 people climbed it. And then after that, lots of people climbed it.
It's like the 4-minute mile, same.
Exactly. The 4-minute mile is exactly the same. It's like, oh, we can't run 4-minute mile. One dude runs 4-minute mile. And guess what? It's like, oh, it's possible. Everybody's running 4-minute mile. Not me, I should point out. Right? Is that it's these same things that look, the statistical models of like what's unlikely and what's likely here is that it's a whole thing of different things that come together. And only when they all come together is when we care about it. So that's when we see it, right? The reason why we are talking about COVID-19 is because it's a pandemic.
It's a freak event.
Right? Yeah. Exactly. And the only reason why we have a pandemic is because, it's a freak virus, right? It would have to be, for example, the furin cleavage site. So it's like Ralph Barrick, for example, has been talking about, you know what? I think furin cleavage sites are pretty important in emergence of coronaviruses, right? Turns out, furin cleavage sites are pretty important in coronaviruses.
And we know that from flu as well.
Right? We know this from the avian flus, right? That in fact, the highly pathogenic avian flu influenza viruses, the hallmark of those are furin cleavage sites. The common cold coronaviruses, while they don't all have furin cleavage sites, right? Many of them do. So it's all these things. It was like, it might seem mysterious. And it is, but it is to an extent, right? It might seem mysterious, but the question you have to ask you is like, yes, but is it highly unusual given that we have a pandemic? And the answer to that is no, actually, it isn't.
I've got to move us on because I want to get to these 4 questions quite quickly because I think that will be a really nice roundup. So the initial response from, let's say, lab leak advocates seems to have been that the papers, since they were preprints, have been completely rewritten. They've been completely watered down. They basically had the shit kicked out of them by peer review, and now they don't really make any declarative statements. And you've got people like our favorite book writers, Jamie Metzel and etc., saying, quoting this line, "However, the observation that the preponderance of early cases were linked to the market does not establish the pandemic originated there and other things. And also sort of crowing that the word dispositive has been removed, which was never going to be allowed to stand in a scientific paper, as you said, because nothing is 100%. So how do you respond to this sort of reaction to the papers that they're like, they're nothing now? We've seen the memes of like, you know, the big dog like coming along in the preprints, and then the like tiny little puppy now for the paper.
Look, the papers have undergone extensive peer review, right? And yeah, we've done a lot of rewriting on this in response to peer review, editorial review, response to new analyses. Some statements that reviewers found were too declarative, for example, that's fair game, right? I mean, in fact, I think one of the reasons why Mike wanted to write the Bloom et al. Letter in Science is because you thought that we, myself, Eddie, Andrew, Bob, were probably too declarative in proximal origin, for example. That's totally legit, right? I mean, I disagree with that. I think we perfectly rounded it, right? But you can have the statement of saying, like, we think it's too declarative. It's like, sure, I disagree with you on that, but I get your point on this. And when you look at the papers, is that you can ask yourself the question is like, yeah, they're rewritten, but are any of the conclusions changed? No, they're not. There is one thing of clarity, which I will say the dispositive and things like that, which actually in the beginning is that there are two things to this paper. One has to do with the epicenter. Where do we really see all of this starting out? And that's very clearly the Huanan seafood market, which to me is the dispositive part, right? And I think most people would agree with this. But then you have the second aspect, which is the emergence aspect of this virus.
So like, all the kind of upstream stuff are still sort of
exactly
open. Yeah.
And that's perfectly fine. If you take the Worobey et al. Paper by itself, does that really conclusively show that, for example, this couldn't just have been an amplifying event? No, that paper alone does not show that, which is why some of the moderation around the emergence aspect of this gets modified in that paper. But here's the thing. There's another study where we look much more specifically at that particular question, right? Which in itself, of course, does not include the Worobey et al. Data, right? Because that's a different paper. So that you can then talk about that. You then say, look, we then bring the papers together and saying, what do the papers actually tell us here? Well, they tell us the same thing, right? And none of the major conclusions, in fact, not even just major conclusions, none of the conclusions have changed in any of the two papers since the preprint stage and the publication stage. But of course, statements have been moderated, statements have been rewritten, papers shortened to make things clearer, new, and I was like, this is peer review, right? And the papers have been under, I think, peer review for 5 months, right? And we had 5 reviewers, I believe, on at least on the Worobey, and I think maybe 4 on the other one, right?
So it takes time.
Did they ask us critical questions? You bet they did. Of course they did. That's the job of a peer reviewer, right? And did all of them, all the peer reviewers, agree with all the statements that we made in the papers? No, of course they didn't. Right? That's not.
There seems to be a weird fight going on at the moment, and I'm interested to see who's going to win between the papers are boring because they're just the preprints from before, and the papers have been completely rewritten and now say nothing. So it's going to be interesting to see like which way Caesar's thumb goes.
Yeah, it's funny because I mean, you even see that in the same tweet, right? You're saying like one statement is like the papers are, well, there's nothing new here. It's exactly like the preprints, and then the next sentence is like they've been completely rewritten and there's nothing in them. It's like, so which one is it? You can't have both. Right. And again, it comes down to, I mean, you mentioned Jamie Metzel, right? And somebody shared that tweet with me. I mean, I should say I don't read these things because it's just, it's mind-numbingly dumb, right? This is a complete waste of time.
We've backed off from a lot of this stuff now.
So, I mean, he's just, he's not, I am happy to talk to people that believe in a lab leak who engage with the evidence and the data, and we can disagree, right? Yeah. That's fine. But when people are completely unable, or unwilling to engage with any of the evidence, I don't
just pointless.
Need to listen to that. It's completely pointless, right? But yeah, Jamie mentions one sentence taken completely out of context, right? And saying like, oh yeah, so now they're saying it's 100% open. It's like, are you just assuming that other people will not read it? Which is,
but I think that is the point.
probably what
It probably is what he's assuming, because most people just want the headline from their favorite political actor, and that's it.
I mean, you see the same thing with my email to Tony Fauci, right?
Yeah.
Where I say that the features possibly, I can't even remember what I said. I said possibly inconsistent with what you might expect from evolutionary theory, for example, right? And people love to like drop the like possible, because that sort of messes it up, right? It sounds better if I just said that it's inconsistent with the expectation from evolutionary theory. Very importantly, though, is that they love to forget about the next paragraph, where I say, however, these conclusions might change, and I even say might change over the weekend. We need to look at this much more closely, and we have
we don't need that part. That's context.
Exactly
boring.
Right? Yeah, because it's boring. Because then all of a sudden, when you take that, it's like,
it's just,
so
normal scientific.
In fact, he changed its mind in the time frame on which he said he might change his mind. And I should make it clear. That very specifically here, I'm talking about the engineering aspect here, right? Because again, there's so many different versions of this lab leak, and there was this tissue culture version of this, which is in fact described in Proximal Origin. And I would say even past where I sort of left like the engineering aspect on the table, because that just does not make any sense whatsoever, is that this idea about, maybe it was actually cultured, that persisted for longer. Right. And I would say we had not completely discarded it at the time of publication, although we were like, you know what, this is just not possible. It just doesn't make any sense given how unlikely all the upstream events would be. Right.
So here's the spicy question, Kristian, as we sort of come to the last couple of questions. And beware of this, because when we did this with Stuart Neal, this became the talking point for the next couple of weeks for Lab Leak. So given the evidence that we have, what would you say is the most probable, even if it's incredibly improbable, lab leak based scenario that's still, within this
possible
universe? Yeah.
That stays within the possible.
Within the physics of this universe. Yeah.
I honestly, maybe the culture based of this, right? That maybe a virus got sampled and they would have to have gotten incredibly lucky just finding it, just to be clear. Of course, they had to have been lying about it too. Got that cultured in the lab, although I should say only briefly. We'll have Joel is leading another study showing that actually the data is inconsistent. It disproves like more long-term scenarios of this. Somebody faffing about and then got infected from that.
So, right. But the virus already had every element. It already had the furin.
I would say it had so basically what we're talking about here is we're talking about a natural virus.
Yeah.
Right.
That took a detour over the lab, basically.
Well, so that's the thing. So that's the problem I have with that, right? Yeah. Because the problem is that when you then say that is that then you accept it's a fully natural virus. So basically what you're saying is that, yes, and I think the most likely way in which this virus came into humans was via that lab. And it just doesn't make sense, right? Because that scenario in itself, given that we know about the animals sold at the markets, right? That in itself is very, very unlikely compared to that. And of course, you have all the actual evidence you then separately have to contend with. There is this separate version of this. People love to bring up this diffuse proposal, right? Which a small part of that talked about furin cleavage sites. And this is all clearly Ralph Barrick's work, right? So the engineering of that furin cleavage site, but that just, the more you think about that, the more you look at that furin cleavage site, the more you look about how it's both evolved and remained stable during the pandemic, is that the engineering aspect of this just does not make any sense at all. I'll say the reason why I was sort of like around the engineering and the culture in particular early on was part the furin cleavage site, but also partly because of the actually more, I would say in the beginning, because of the receptor binding domain. because the receptor binding domain really was unusual in some aspects, right? It had aspect of the SARS-1. It didn't look like it was a specifically designed one, right? But it looked unusual. But then you find it in a pangolin coronavirus. You're like, oh, there it is, right? So clearly that exists in nature, right? So that doesn't make any sense. So I'll say, yeah, for me, it's the culture one, right? But I find it, hugely unlikely, because of the actual evidence that we do have.
Why do you think, Kristian, that the Chinese CDC comes to a different conclusion about the market with the same data set? That's
yeah, I mean, that's the unfortunate
question.
aspect. I mean, I think this is really unfortunate. And I would say, like, the papers we just published, in fact, we shouldn't be the ones publishing these papers. All of these papers should have been published back in 2020 by Chinese scientists, right? It would have been great if we had been part of some of the, more careful studies around molecular clock, for example. But frankly, these papers should not have been published by us. They should have been published by Chinese authors. And they should have been published in 2020. And the reason why I'm saying that is that, look, if you are truly just interested in understanding the emergence, aka the origin of this virus, which is what I'm interested in. I'm interested in understanding, like, how did we end up with the COVID-19 pandemic, right? That's my question. That I would hope would be a question that all scientists would be interested in. So if it was just down to the science, well, then that's the question. Everything else does not matter, right? The problem is that this changed very early in the pandemic, where this is not about understanding emergence. This is about blaming somebody for the pandemic, very specifically here, China.
And I guess that's why.
And more recently, the US.
And now more recent. So that's the problem, right?
Because I guess that's why Mike had that regret earlier in the discussion, potentially,
yes,
which I was kind of sort of trying to nudge him on of.
Yeah,
did that make them sort of draw back when it was like, right, give us everything, we want to see everything? I imagine, like, I always think about what would the Americans say if the Chinese said, right, we want to send someone in your lab, show us all your books. Like, it's just not going to happen.
Right. Yeah, it's not. And look, I don't disagree on the message. Like,
no. We want to see everything. We all
right?
want to see everything.
We all want that, right? I also want chocolate. I want chocolate all the time, right? I really do. And Danish licorice, right? But I can't. That's not good for me, right? So even though that's what I want, well, I can't have it. Right? Well, maybe I could, but then I'll probably die. So the thing is that when we're talking about these things, right, is that we need to realize there's a set of things that we all agree that more investigation is required. We all agree that better data is what we want. We all agree that we want more data. The question comes down to, yes, but what can we hope to get?
And how do you get it?
Right? And how do we get it? And the problem here is that this is what I was so dismayed by the science letter, right, is that was the process the WHO had started. And I actually think they achieved quite a lot. Did they achieve everything? No. Did you expect them to? No. Right? Does it mean that because they didn't achieve everything that it was completely useless? No. The problem was in the science letter, and this is what, I mean, I mentioned it when Mike was on, right? And Mike and I talked about this a lot, is that this problem of saying, like, look, we have no evidence whatsoever. And in fact, the evidence we do have has been produced by Chinese authors, so we can't trust them. It's like, well, if that's your standpoint, we move from nowhere. Yeah. And then you turn into the last aspect of that paper, which is we want everything. And I'm talking literally.
Yeah,
we don't
trust you. We've got our misgivings, but we want everything.
Exactly. We want everything. We want all of it to be verifiable and things like that. That's not to engage in a conversation with people trying to figure out what the emergence is. That is simply just grandstanding. And very specifically, it's grandstanding from American scientists, most of whom have no expertise whatsoever in addressing these kinds of questions, none of whom I think have any expertise in generating the vast majority of the data we're talking about here. It just turns into grandstanding. That is not, from my perspective, that is not a helpful way to engage in this conversation. And that's what I was so upset about. And frankly, that was because Jesse Bloom's paper preprint has also come up lately, right? That was the aspect of that I was so upset about too, because you are accusing people of obscuring the trusted structures of the science or whatever. And at the same time, it's like, yeah, and I want all your data. Not acknowledging, you know what, the data you did your analyses on, there, it was already in the paper. It was in table 1. That's why your table 1 is basically their table 1, right? Instead of engaging in a conversation saying, look, I just discovered this data. It was in a table, so it was not very accessible, but I took this data, I made a paper out of it, and I thank the Chinese authors for having created and released this data in the first place. That might have been a useful way in which to engage in this conversation. Then people like me could have come up, because I have lots of technical issues with that paper too, which I talked about.
But you've got that relationship now, and you can start
exactly.
making collaborations.
At least you have a starting point, right? If you come to people and you're saying, you must give me all of this, I mean, do you think it's like, yes, sir, of course. Here you go.
To be honest, I have to disagree, like on the role that you think scientists ever had over this.
Oh, sure.
I think, the problem was that, once you mix science and politics, all you get is politics. And this was the problem from the beginning.
Yeah.
And scientists,
the Trump China
thinking about, now maybe the
kind of
wording was not correct, or this was too strong. And these are all, marginal sideshows. And,
I mean, it still is important to clean our own house as much as we can, but even
of course, but you can be the most perfect scientist, and probably we would still be in this situation where, Chinese people use the welcome propaganda on their own people, and the Americans love blaming China for geopolitical and domestic political reasons. And, everything is a right-wing funnel towards conspiracies. I think there are larger forces at play.
And again, from my perspective, right, this is why none of this is important to me. People love to bring up, it's like, oh, but Trump was the president at the time. It's like, that doesn't matter, right? I just care about understanding emergency.
Yes.
Who's the president is not a concern of mine. What I will say, though, and this is important because these things get mixed up, and it's like, oh, so you just trust everything that China says. It's like, no, I don't. I'm not stupid, right? But I'm not talking about what China is saying. I'm talking about data generated early in the pandemic from Chinese scientists, right? Who I do, to some extent, trust. And very importantly, it's different scientists, it's different kind of data sets, it's different kind of stuff we can look at, right? And then really importantly is that when you are saying, look, I don't, for example, Xu Shangli, right? She comes up all the time, is that there's no version of this, that if you say that this came from her lab, you are also saying that she is lying, right?
Yes.
There's no other version of this works for you, right? You have to, if you want one, you have to get the other. And what's really important here is that if we actually had evidence to suggest that indeed she did, well, then unfortunately we would have to say so, right? The problem with the message, for example, in the science letter,
you're making
here,
the accusation
more broadly
without having anything.
Exactly, you make those accusations, sort of bypassing the thing like, yeah, we're talking about specific scientists, but that's not what I'm saying. No, that is very specifically what you are saying, and very specifically, it's unfounded speculation because you have absolutely no evidence.
And to be fair, this is what I see a lot on Twitter from our favorite book writers again. They have this very they say it in nice words and friendly words and a calm way.
Yeah.
Very, very cutting and very, very serious accusations about what people have done and how they've been responsible for the deaths of millions of people, and how they're quite happily covering that up. And because it's said in nice, calm words, it's accepted as reasonable. But it doesn't help us get to the bottom of the issue.
No, it doesn't. Well, it doesn't help us get to the bottom of the issue. And really importantly is that it still remains unfounded speculation, right? Because there is no evidence for any of this. And I'll say, there's a Vanity Fair article recently, right, which a lot of these issues came up because I was very specifically talking about, Jesse Bloom's preprint at the time and how there was a conference call and there was disagreement and stuff like that. And it's like, yeah, of course there were disagreement on that call. I was shocked by the statements made against scientists in China that had generated the data we're talking about. Very specifically talking about how they're abusing the trusted structures of science, how they were deleting data to obscure the origin and things like that. Yeah, I have issues with that because, again, I don't think that's why they deleted the data. In fact, the explanation for why they deleted the data, which I have tried to generate this kinds of data, right, more than 60,000 by now, is perfectly reasonable. So maybe we should just go by that. And really importantly, too, is that at the end of the day, the data was always
there. Yeah.
Right? It was always available in table 1. So even if you think that deleted these BAM files or SRA files because, you know, they wanted to obscure the origin, actually they didn't, because they had table 1, right? So that's when I have like, so me personally, I do actually have an issue with that. And I get frustrated when I try to explain the issues around this. This doesn't mean I'm an apologist to China. It doesn't mean that if we actually had evidence that they deleted this to actually obscure the origin, right? Is that if we had that evidence, of course, I would be the first one to say so. The problem is that we don't. And it remains entirely in the realm of conspiracy theories, speculation, that conjecture. There's just no other way of putting it. Then you add to that what I find to be problematic, analyses too, some very, very simple statements, right, that I just disagree with because, well, I've been doing these kinds of analyses for a long time. And again, we do them a little bit more carefully. Bring that up too, where that's the scientific aspects of and issues I have around this, right? But they're separate issues, right? And I do think it's problematic that if we really want to try and understand more fully the emergence of this virus, we got to focus on the evidence. And we got to focus on how can we get back to a stage in which we can actually work together with, for example, Chinese researchers and Chinese authorities. That's going to be harder, but let's try with the researchers first, right?
Yes.
So we can understand more fully the emergence of this virus. Because these papers are not the end all be all on the emergence of this virus, right? Because while we now have the focus of the market, we don't know upstream.
So that's my final question, Kristian, because we've taken a lot of your time, and I want to be mindful of that. The final question I have, and I don't know if Philipp has anything else. I mean, I could go on talking about this for 4 hours, and I've got a load of other questions, but I think we've covered the most important ones. What are the main things that are still big open questions that remain to be answered on the SARS-CoV-2 origin? So you talk about those upstream things, and lab leak advocates have been making a big show of this on Twitter. Oh, they don't know these upstream, so it could still be a lab leak and blah, blah, blah. What when you say upstream, what do you actually mean by those aspects? And what do we still want to nail down?
Yeah, so to the question of have we disproven the lab leak, right? No, we haven't. We never set out to do that. What we have shown is you look at all the evidence, and as we have said previously, the lab leak hypotheses are just not viable. They're implausible, right? Not impossible, right? So we should still keep an open mind in case other evidence were to come to light and then address that evidence. Critical. Any scientist would agree on this. But really importantly is that you look at all this evidence, it points straight to a market in Wuhan. It looks an awful lot like SARS-1, but not identical to SARS-1. And really importantly, this time, this small outbreak happened at a wet market. We ended up with a devastating pandemic that is going to be part of the rest of our lives. Right? COVID-19, or SARS-2 rather, is now a virus that's going to be with humans certainly for the rest of my life. Right? So we got to figure that out. Saying with SARS, it worked out. With SARS-2, it didn't work out. Why? And we don't know the answer to that. We know that, they are associated with wildlife. They're associated with selling of wild animals that then go into the human food chain here. We know that, but we don't understand the upstream events. What animals specifically are we talking about? Where did those animals come from? What are the risk factors and where did those come from? How is all of this connected? Why did this happen in Wuhan specifically? And this is one question that people either Wuhan has to be the most likely one, otherwise it wouldn't have started there, right? Which is obviously complete rubbish because these things try to start all over the
survivorship bias.
It's got to be exactly the survival bias, right? It's got to be somewhere. But then also the idea that Wuhan is a hugely unlikely place for this to start is actually not true. Very specifically, for example, we know that they got wild animals from Hubei and nearby provinces, right? And in fact, we know that several of those farms were positive for SARS-1 as well.
Yes.
And in fact, if we look at those viruses a little bit more, it looked like, well, maybe actually they could be, basal to the outbreak, or the SARS-1 outbreak, I should say. But that requires much more careful analyses, which we're doing now. And I should say it's very, very difficult. In fact, I thought it would be easy to figure out. It's very difficult. So we might never figure it out, right? But you have the components all there. So Wuhan itself, but it might not be the top place. That would probably be somewhere in South China, right? It might not be the top place. It's that maybe it is in top 10. And of course, we need to understand all these factors. And currently we don't. And the reason why we need to understand this is that, well, the next time we can minimize the likelihoods of these outbreaks by maybe more control and things like that, right? But even that is difficult. So we just have to accept that, look, these kinds of events are going to happen again. What we really need to focus on is the next time they do, this whole thing where that outbreak turns into a pandemic, let's make sure that doesn't happen. And we did that again successfully with SARS-1. I do hate to think that I actually do think that we could have done the same with SARS-2. We can't now, right? Just to be clear. But in the really early stages of the pandemic, with more concerted effort worldwide, I mean, China managed to actually extinguish the virus, right?
Yeah.
Is that actually thing that it could have reasonably been done? We just didn't. Right? Because we never actually tried seriously, right? We completely we don't need to test for this virus as a basic level, for example. So that's why we need to understand. We really need to understand everything upstream. And I think we can, right? I think that while we might not be able to get all these aspects, maybe we will never be able to get infected animals. In fact, I find that highly unlikely at this stage because farming and everything was specifically shut down, right? What we can do is that surely there are people that have additional knowledge around the early events at the Huanan seafood market, that have additional information about what animals are we talking about exactly, like all of these things. Exactly, where did they came from, how were they distributed across markets, whether other markets, like whether early events, like all of these things is absolutely within the realm of what we can investigate and hope to figure out. But of course, it goes back to my first point, which is that we can't do that by looking at EcoHealth Alliance emails, right? We need to work with China. We need to work with researchers in China to figure that out. And we do so because we want to understand the emergence more fully, not because we want to blame this pandemic on China. And of course, the corollary is that China is now blaming the pandemic on the United States, right? And then you get nowhere. Because that's their conspiracy theories, is that it came from a US, bioweapons lab, right? Which is just as ludicrous.
And they have also, it's very interesting. I talked to some of my Chinese former colleagues, and they said, the lab leak thing is like, it's a mirror image of what happened in the US. Oh, yeah. How it started there. It started with some random bloggers, talking about bioweapons. Then it got specified via cold chains and via, work that was performed. And now, you have, speakers like Jeffrey Sachs, rehashing these fundamental Chinese property that it was the University of North Carolina because, they were doing the research on coronaviruses like the in the US people say, at the WIF they did the coronaviruses, and they say, in North Carolina they did the coronaviruses.
It's like a sort of shady sort of reflection of what we see.
That's the problem. This is why, from my perspective, it's like, just stick to the data, right? Just, realize that you need to collaborate to figure this out. Realize that you're doing this because you want to understand emergence more fully. It's the only reason why you do it. There's no other reason, right? For me, there is no other reason, right? That is the reason. We investigate emerging infectious diseases. I lead research centers on exactly that topic, right? So, of course, we're interested in understanding the emergence of infectious diseases because that's what we do. So do that realization first, realize that it's necessary. Then let's figure out how we do that best. And I will say WHO SAGO is not a bad way of furthering some of that science. But the point that Mike made earlier, which is that, yes, but a lot of these analyses will have to be done by specialized experts. We are some of those in a particular area, but there are other versions of this in which we are not, right? And of course, those scientists need to be able to do that research. And to be quite frank, like right now, that is very, very, very difficult, right? Almost impossible. And it comes with a lot of personal cost as well. I can tell you that. right? That aspect of it is no joke. And I don't think that's really where we want to be, because at the end of the day, it makes the world more unsafe. I should add that if you want separately, you want a conversation about how we need to restrict gain of function research, how we need to rethink how we grow, for example, coronaviruses in BSL-2, which is not just in China, this is across the world, right? They said, sure, of course we need to have that conversation, because we now have a pandemic that is caused by coronavirus. So we need to reassess.
And I think people don't realize this, that the scientists who are saying it's a zoonotic jump, it's not here and here. They also say, like Stuart, we've talked to Stuart Neal a lot, and we said, look, do you think we need, do we need more oversight of these labs and what's going on and potential gain of function that's going on? And he's like, yes, definitely. Drastic has found some really bad work that's been going on, and we need some regulation with teeth to sort of work out and have more kind of oversight on this. So nobody's sort of just
nobody's disagreeing on that.
Zoonosis, right? We can't do anything about lab safety. That's just nonsense. Like, people have been talking about this in the field for years. I remember going to a talk with Mark Lipsitch in Cambridge in like 2016 or something, or watching it on YouTube. And this has been discussed for a long time. There's a moratorium on GOF in the US, I believe, until recently. So it's not something that the field is unaware of or takes lightly.
No, it's absolutely not, right? And I think it's just that what I do object to is that you sort of mix them all together and say it's basically the same conversation. It's not, right? These are very, 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 very separate conversations. If we knew or had reason to believe that the COVID-19 pandemic started as a result of a lab accident, that would be relevant, right? But here's the thing, the data tells you, you know what, didn't, right? It's just a continuation.
But it doesn't mean that you can't do that lab stuff. And I actually think it's a really dangerous strategic mistake to tie the idea of having this regulation to the specific emergence of one virus. Because then if it goes down in smoke, like it looks like it's doing, then people will turn around and go, oh, well, what were you worrying about? Like, oh, it didn't come from a lab. And there's no need to do that. It's a very silly thing to do, in my opinion. I can understand why people are doing it and using the emotion and the impact of this pandemic, and it's very raw and visceral and right in front of us. But I actually think long term, it's the wrong thing to do. To tie something generic to the specific outcome of one thing, I think is a poor way to proceed.
Yeah, I think it's unhelpful, right? I mean, it does have to be clear, though, that the COVID-19 pandemic itself informs us here, because as I mentioned, is saying, look, this is a coronavirus. And in the past, typically this type of work isolating these kinds of coronaviruses was approved under BSL-2, typically under what's called BSL-2 plus, which is basically BSL-2 plus extra precautions that you would do in BSL-3. My lab, in fact, is approved for this work, right? We have worked on isolating, never successfully, but isolating viruses from bat samples.
Do you have a dentist office? Do you have a dentist office as well, Kristian?
We do have a dentist office as well, and I can guarantee you it looks pretty different than our lab. And, this idea that, there are safety aspects and we only know about this now, it's complete rubbish, right? The reason why I was concerned about a lab leak is because I knew about the work being done at the Wuhan Institute of Virology, including the work being done at BSL-2, because it was published in peer-reviewed papers. That's not something we have found out because of foyers of EcoHealth Alliance grants and reports and emails and stuff. It's complete rubbish, right? All of this has been known all along. So, but again, really importantly is that these things are separate conversations. And because you're saying, let's not mix them up, it doesn't mean that you're saying, let's not have one of them and not the other, right? And it's the same with proximal origin, for example, saying they never wanted to investigate any further. Rubbish. In fact, we actually talk about it in the paper, right? It's complete rubbish.
You get another paper if you investigate some more. That's always good for the science.
We will not, and I will say, we're not investigating anything. We're studying, right? Because I'm not an investigator. I'm not a spook, right? I'm a scientist, and before I was a scientist, I was a postman, right? So I do this because I'm interested in understanding more.
He always delivers. That's,
there you go. Mike actually used to be a forest firefighter, I believe.
Oh, wow.
So,
Cool.
Mike did something cool in his previous career as well. Like, we just do this because we're interested in the question, right? And this idea that there's a preferred narrative, there's a way in which we all want, is like, it's complete baloney, right? And it's just conspiracy theories. And it's very unfortunate because at the same time, you're actually hampering us getting a better understanding what's going on here.
I think that's
this is not helpful.
I think that's a really, really good place to leave it on that message. Unless Philip, you have anything else?
Usually I try to end with a bit more positive notes. So
please
Kristian, can you please lay out to all the people that are worried that nothing has been done, that SARS-CoV-2 is going to come, what can we do, what will be done, and how likely is it that it's going to happen exactly as it did now with SARS-CoV-2,
and then we will let
how
you
can
go.
we stop
we will
it?
let you go then in the next 5 minutes, we promise.
Yeah.
Well,
positive note.
well, positive. Big smile.
You get Kristian Andersen on your post podcast, and then you ask to be positive. No, look,
what is a positive vision? Not a realistic one,
look,
a positive one.
People that follow me on Twitter, people that that listen to me at times, it's like, oh, you're such a negative person. And the thing is, I'm not actually. I'm actually a relatively optimistic person. I just happen to be Danish. But look, here's the thing: when we're talking about something like
Russell Brand's line was very good about you, to be fair. Like I hate the guy, but he said something like the latest fairy tale from Hans.
Hans
Christian
Christian.
Yeah,
that's a good one.
It was
and
a
I'll
good
say
line.
someone like I've seen like Drastic have made some animations and stuff like that, like me deleting emails and stuff like that. I love it.
I just Stuart Neal like dressed in a potato sack.
Oh yeah, it is,
and he's like absolutely stacked. He's like,
Stuart was really buff. I was like, this is amazing. Like, if you could just put those talents to some other things, but good laugh. Anyway, so the thing is, when we're looking at the COVID-19 pandemic, what happened here is not actually mysterious, right? Many people, Shishang Li, for example, Peter Daszak, Ralph Baric, many others have warned about the likelihood of coronavirus pandemics, and we should really do something about it. This has been, gospel for 2 decades now. People like myself have been talking about, look, pandemics are going to happen, so we really do need to prepare for it. And fact is, we didn't. Here's the thing. This is going to happen again. But the emergence event here is going to happen again. But next time, maybe let's not have that outbreak turn into a pandemic. And that is fully within our capability, because what I'm talking about here is not complex rocket science that requires all kinds of technological solutions, right? No, actually it doesn't. It requires, straight up diagnostic tools, for example, having those available, collaborate in a global health aspect and saying, look, pandemics are a problem for the whole world, so maybe we should all try and work towards them not happening. But that requires collaboration, cooperation, openness, all these different things. And I think we need to try and find a way in which we can do that. And I think while the WHO is not the perfect organization to do that, I think they're well positioned, and I don't think there is a perfect way to do this, right? So I think if we can just support that and get back to let's just focus on what's important here, I actually do think that we're in a better place in terms of preventing or minimizing the risk of future pandemics.
I think this is what we always talk. We talked about this with Angie as well. The sort of One Health approach that, what people are doing at wet markets in China, or when African cities are expanding into ecosystems of various animals, or in Brazil when they're cutting into the Amazon and going into these ecosystems as well. All of these things that are going on in these countries, particularly developing countries, which we might think of as, oh, we don't really care, so, whatever. It can have a huge impact on us back in the West. So even from just a selfish perspective, not even thinking about the well-being of these people, we need to understand where these viruses are coming from, educate people about where they're coming from, try and head them off if we can. I think we've probably been quite insulated here in the West to a lot of these problems that people deal with in China and particularly in Africa on a more daily basis.
Yeah, and I think, again, it also needs to be clear, right, that this is the collaborative aspect of this, right? It was like, you say, oh, you shouldn't burn down the rainforest because that's bad for us.
But they're poor and they don't have anywhere to live. And
Exactly. It was like, but you need to understand why this is happening, right? And then we need to all work on a solution to maybe, minimize the risk of this happening in the future. It's the same with the wet markets, right? People are like, oh, that's disgusting. That's like, it is no different than, for example, pig fairs in the United States, right? And factory farming and all these different things, right? Is that it's culturally just incredibly insensitive, and frankly, it's unhelpful. And that's why we all need to come together for these solutions, which One Health is a good way of looking at this. And I'll say the collaborations we do, which are very international, I have many colleagues in Africa, right, is that has been what we have focused on for many years. Open data, One Health approaches, just focusing on doing good science and actually making a difference. I think that's what we need to do. And I think we are entirely capable of doing it. We're just not really moving in that direction right now. And I guess that's not finishing on a positive note, right? I think the positive aspect of this is that I think we can do it.
Yeah.
The negative aspect is that I don't think we're currently doing it.
But there's a positive scenario that we can choose if we want to. I think that's
oh, absolutely.
I think that's a nice place to leave it, Philip.
So a better future is possible.
That's right.
Exactly.
Entirely within our grasp. Yeah.
Kristian, thank you so much. Thank you to Mike, who's not here anymore, but I'll say it anyway. Really appreciate you taking the time. We've gone for ages. I honestly I could chat about it for 5 hours and
we'll do it in the pub next time.
Well, that's a very, very good idea. Thank you again for going through that with us. Have a wonderful evening, and I guess we'll chat the next time something big comes up in COVID origins or something related.
Sounds good. Looking forward to it. Thanks, guys.
All right. Thank you very much, Kristian. I really
Ciao,
appreciate
Kristian.
it.
Cheers.
Bye now.
Bye.
Bye.
I want to know what you think, because you're the scholars of enlightenment that I do this for. So please take a moment, if you wish, to let me know down in the comment section. And if you like this video, please consider leaving a like, subscribing, setting up notifications, and sharing this video more widely. I can't tell you how much these simple actions help me out and how much I'd appreciate it. Thanks for watching. Thanks for being scientific. Thanks for being bad.